Avvio registrazione.
Sì, adesso arresta la trascrizione.
Okay, so Anton, can you hear me?
Yes, I do.
Okay, thanks. So I'm very glad to introduce you Anton from Copenhagen University, and he's an expert in metagenomic microbiomes, and is going to complement the lesson from Marco from this morning. But I leave the stage to Marco, who's collaborating with Anton already a long time ago. So please, if you want to introduce Anton to me, thank you.
Thank you, Stefano. A very short presentation, so we can start the class after that. So Anton is associate professor at the Globe Institute at the University of Copenhagen, and he is a coordinator of the H2020 project, 3D-Omics. He is a coordinator of the Earth Hologenome Initiative, and is also a scientific manager of the H2020 project, Holofood. We know since some years, and in the last year we were working together, and so on different field in microbiome research, in particular trying to understand how microbiomes can complement the host biology. So, but anyway, I will leave the floor to Anton now. So, Anton, please, you're free to start.
Excellent. Thanks a lot, Marco. Can you hear me properly? Yes. Okay, cool. I will try to share my screen and try to find out what's the best way to have the optimal setup so that I can see you and you can see the slides. Can you see the slides? I know it's not in presentation mode, but otherwise I don't know if I will be able to see you. So that's the reason that I am.
Anton, is very small.
It is very small.
Yeah,
okay.
Yeah. Maybe two.
Yeah, it's better the presentation mode.
What about now?
No, it's still small.
Still small. Okay, let me then change the strategy. Let's go for this other. I have two alternatives. So then I can go to the.
Wait a second. Yeah. Perché come faccio a farlo? Non riesco a governarlo, capito?
No, lui deve condividere lo schermo.
Perhaps, Marco, can you flip the computer so that I can see myself what you are seeing and try to.
Yes.
It might be easiest rather than you describing me. Okay, so if I do the slideshow mode, is that better?
Yeah, this is better.
Okay, and this is okay. The problem is that if I go for the slideshow mode, I am unable to see any of you.
Yeah.
So let's see if we can find an intermediate solution. Let's wait a second. Okay, so you can you can see only part of it.
Yeah, not all. Yeah.
Is it possible? Can you extend the so make me smaller and the main screen larger? Would that be possible? Or
wait a second.
Or if I enter into full screen.
Non riesco a farlo perché è bloccato. Sorry, I'm speaking Italian.
Capisco un po'.
Però cambia poco, che non riesco a tirare su questo. Cioè. Perché non riesco a non vedervi. So this is the best I can do.
If I can. Okay, I think it should be good enough. Is it big enough, large enough for you, for reading what's written there?
Yeah, it's okay.
Is it okay? So that'd be good for me because that way I can also see the students, and otherwise I won't be able to see anyone.
They have their device, so they can see you by using their mobile. So please, can you stay on the left side of the room? So here on the center to the left side. So just a second. Okay. So if I leave the microphone on, is okay for you, or you see?
Yeah, it's completely fine.
Okay, so I will leave it on.
No? That's perfect.
Switch off the microphone. Okay, in the room. Thanks.
Okay, I think we can start now.
Excellent.
You can also follow the lectures with the headphones. That's the best for you. Following, you have your own headphones. It's the best.
I think that would make a difference with the microphone mainly. Yeah, for me to listen to them, but
Wi-Fi is
we can try.
with the Unibo account. You all have Unibo account. No, you must have your name at Unibo. That's your account, the password. Everyone has. Otherwise, you're not a PhD student at university.
Okay, so we can start? Okay, Anto, we can start now.
Super. Excellent. Thanks a lot, Marco and Stefano. So I hope the setup will be good enough. I know it is not ideal, but ideally I would like to interact with you along the session. Otherwise, it's going to be too boring. So yeah, hopefully we will manage to do that through this setup. So just to follow up on what Marco said about who I am and where I am coming from. So I am one of the principal investigators of the Center for Evolutionary Hologenomics as well, which is a center of excellence of research that was launched 2 years ago. And what we do is to study animal microbiota and plant microbiota interactions using multi-omic methods. That's our topic of research. And in my personal case, I do that on animal microbiota interactions. So I don't do plant research. I must admit that fish microbiota research is not my main topic of research, even though we have a few projects on that topic. And that's my only connection to marine organisms or to water living organisms. I must admit that my bulk research is on the interactions between vertebrates, terrestrial vertebrates, and their gut microorganisms. But just to finish about myself, I am leading a research group of 16 people, and we are running a bunch of different projects that always within this idea of animal microbiota interactions using very different approaches. We do a lot of multi-omics. We also are starting now working on gut-on-a-chip organoids. We run animal experiments. We go to the field and we work with wild animals. So it is quite diverse in that sense. And today I will try to show part of that diversity, even though my talk won't be focused on research activities that we carry out, but it's going to be more theoretical technical. But if you have any questions, I will be very glad to answer. You can find my email address in the slides. I guess Stefano and Marco will share these slides with you or the recording. So you're very welcome to contact me anytime. This is how I have designed the session for this afternoon, evening. So hopefully we will have 4 sessions and some breaks in between. to make sure that we don't die. I guess that by 6:30 we will be all super tired. So I will try to keep this as dynamic as possible, but I cannot really promise anything because I know it is a very long day. So I would like to ask you questions, and actually many questions, and I would really love to get some answers. I will not be expecting you to be experts on the topic, so the aim of the questions will not be just testing your knowledge, but rather will be reflecting on some ideas, and that means that in some cases you will have some. This was a GIF actually, so it was supposed to be moving, but not in the PDF anymore. But I just added this image to show that perhaps some questions will not be very easy to answer, and there might be some uncomfortable situations. I will also let you have some silence periods, and I think this is mainly for you to think about the questions. So there will be also some moments of, well, I don't know what to do. And but I think that should be part of the process. And so I will ask many questions. I will wait for some time, and if any of you is willing to respond, that would be great. Otherwise, we'll see what's the best way to create an interactive session. So just showing this agenda for this afternoon, we will start with the first session, which is an introduction to animal microbiota interactions. I know that Marco has given you a few hints about this topic. In this case, I will try to deepen a little bit more, and this will be the introduction to the topics that will come later. And as you know, the marine biodiversity is extremely diverse. And as I mentioned, I will be mainly focused on animals because that's animals and their associated microorganisms, because that's my main topic of expertise. I don't really have much knowledge on other domains, and so I won't be covering algae. I won't be covering any other plants, etc., etc. So just so you know that it's going to be focused on animal microbiota interactions and mainly vertebrate microbiota interactions. But many of the topics we will cover will be also extrapolable to other host taxa. So the first question I would like to ask you is, what do you think an animal is for a microorganism? When we are thinking of these systems where we have some host and some microorganisms that are associated with that host, what is an animal for each of these microorganisms that live in or on the surface of the skin of the animal? Have you ever been wondering about this? Would you have any answers for this? Would any of you like to respond? There's not just one answer. There are many answers.
The environment.
If I heard properly, it was the environment. It would be nice whenever you are to respond. Can you raise the hand, and then I can see, because I don't know otherwise who is talking. Can you repeat this? Yes, I do. So you said the environment.
Yes, I said the environment because they use it and they just see animals as resources to use. Yeah.
Okay, thanks. Would any of you add any other issue related to what an animal is for a microorganism? What can it be? Could an animal be also the source of nutrients, for instance? There are many things in the end that could be. So could be the environment or the habitats, could be the shelter, could be the source of nutrients, and there are many other things that how we can define what a host is for a microorganism. But we can also flip the side of this system, and we can also ask, what is a microbe for an animal?
Maybe the animals don't care that.
Can you raise a hand or stand up? Yeah.
I think that maybe the animals don't care about the microbiome.
I'm sorry, I wasn't able to listen that properly.
I mean that maybe the animals don't care about the microbe. That's fair.
I'm sorry, that's the animal. I wasn't able. Can you repeat that, Marco, perhaps? Closer to the mic.
Said that maybe the animal don't care about the microbes.
Don't care. Okay. Good. That's a possibility indeed. But will you have any other thoughts about what a microbe can be for an animal? There is an obvious one that historically microbes have been linked to. Can you think of anything? Well, a microbe could be a pathogen, for instance, right? It could be detrimental for the host. But could a microbe also play another role for a host? Don't be shy, come on. Otherwise, really.
It might be a source of energy.
Could be a source of energy, could be providing some kind of means for getting some energy. Yes. How? How could provide energy a microbe to an animal? Yes. Yes, I wasn't able to hear that. I'm sorry.
He said digesting.
Digesting nutrients could be. Yeah, so there are many functions that microbes can play. So it could be a source of nutrients. It could be a pathogen, but it could be also, for instance, a stimulus for starting a physiological process. It could be a stimulus also to trigger different kind of processes during the development of the animal. And there are many other options in this case. But the main message is here that I would like to transmit is that hosts, this system of hosts and microbes is not symmetric at all. What hosts are for microbes and what microbes are for the hosts can be very different and is very different. There are some elements that are in common, but there are many elements that are not in common. So this is one of the main ideas we should get. So this asymmetry of these systems, and this asymmetry will have huge implications on how we analyze the host microbiota interactions and what kind of consequences these interactions might have in ecological, in evolutionary, and in applied processes. And this is actually the main topic of this lecture, which is the interactions. So we are talking about animal microbiota interactions, and I will be underlining this word, which is the interactions. I am, of course, interested in animals and microbes, but I'm mostly interested on the interactions. So this would be the next question for you. How do you think animals and microorganisms interact? What would be your thoughts on this? Do you have any idea how these interactions could happen? Or at which level these interactions happen?
Mostly through chemical exchange.
Marco, would you echo this? So perhaps say it's very difficult.
She said mostly through chemical exchange.
Excellent. Yes. Do you have any other thoughts? There's this possibility they are interacting through chemical exchange, through biomolecular processes. And could you think about any other level of interaction at a different scale? I have included 3 different scales. So one is the bottom one is, as you mentioned, these molecular interactions or biochemical interactions. There is an exchange of biomolecules. There is a production of molecules from the host, a production of molecules from the microbial perspective, and they are able to sense each other and to trigger different processes based on these molecules. But these then have really huge implications at a larger scale, because there are ecological processes that are affected, and there are also evolutionary processes that can be affected. So these interactions between animals and microbes can be analyzed at very different scales. I would say the obvious one and the let's say the one that occurs at the highest resolution is this actual mechanistic way in which animals and microbes are able to sense each other and to react and create different kind of processes. But then this can also be analyzed at a larger ecological framework, and even in a larger evolutionary framework. And we will be covering these 3 scales of the interaction throughout this lecture. Something that is important to consider is the resolution at which we analyze host and microbiota interactions. I have added this figure from a recently published paper of ours, where we show some examples on how complex the animal microbiota interactions could be, or the microbiome associated with an animal can be. And I will follow up with this to show what is the importance of how we define a resolution. So here in this figure, you can see that there is an extending gradient of the complexity of the microbial community. And this will, of course, have an impact on how we analyze these interactions. But most importantly, you can see this example here of the bobtail squid light organ. And here in this figure, it is shown as a one-to-one almost interaction or relationship. We have a squid and we have one bacterium. And this is true when we focus on the light organ of the skins of the squids. So when we go and focus on the light organ, there is just one symbiont, a Vibrio fischeri, which is the one that enables this animal to have this production of light that's by itself, the animal by itself would not be able to have. But if we zoom out and we consider the entire animal, then there are many other communities that can be also considered. There is the skin community beyond the light organs. There's the gut microbial community. There will be the oral microbial community, different sections of the intestine, etc., etc. So whenever we are studying animal microbiota interactions, it's extremely relevant to define properly what is the resolution, the spatial resolution at which we are analyzing these interactions. This is another example of a just published article. So this is on a PhD project that we are conducting on marshland fish in the Mediterranean part of Spain. And what we did in this case was to compare the microbial community from the digesta of the animals and the intestinal wall. So when analyzing fish and many other animals, we can have different strategies to collect microbiome samples. One option is to digest the entire intestine, for instance. Another option is to flush out the intestinal content. Another option is to grasp the mucosal barrier. And depending on what we do and how we get the samples, the microbial community we will characterize will be different. So this will also define the resolution at which we will be analyzing animal microbiota interactions. This is just an example of that study and the result of that study, where we analyzed 3 species, Aphanius iberus, Gambusia albrucki, and Valencia hispanica. And by comparing these pairwise comparisons of the digesta and the intestinal wall samples from the exact same individual, we can see that the intestinal wall contains a considerably lower diversity compared to the digesta. And this happens in most of the cases, and it is statistically significant. So depending on which sample type we use, we will be characterizing a different community. And this will, of course, have a huge impact on how we characterize these interactions. Of course, we have different options. We could go for just for the digesta, just for the intestinal wall. We could also mix both of them. But making one choice or the other will have a huge impact. I would like to ask you now whether you would have any a priori decision on which sample would be most interesting to analyze host microbiota interactions in this context. Would you go for the digesta sample, or would you go for the intestinal wall sample or mucosal scrape sample?
An idea of which sector, which compartment is more appropriate for microbiome survey study, which you like to choose?
I have an answer. I have a question that maybe you can partially answer.
Okay, thank you. Take out the mask and speak out.
I have my microphone.
Ah, okay. Oh, yeah, also use the microphone. It's even better. Thank you.
I try to speak with my microphone.
That's perfect. I can hear
the question is how much the time from the death of the animal can influence the microbiota, and if the effect is the same in the digesta or the intestinal wall.
We haven't empirically tested that, and we know that there is a change in the microbial community. But in this case, let's assume for this question that let's say the distortion of the microbial community is identical between the digesta and the intestinal wall. I must also say that the samples here were taken about 1 minute after euthanizing the animals. It was very fast. We euthanized each animal and then dissected, and the samples were taken straight away. So we should not be expecting any bias in that sense.
Thank you. So I don't know. I was expecting a difference.
No, but it is a very good question, and this is something that I didn't include in the slides, but it is extremely relevant, which is what is the let's say the representativeness of the community, the microbial community that we sample, compared to the biological community that we are trying to characterize. Because it's not the same to sample a fecal material where we are trying to infer something about the lower intestine. It's not the same getting a fecal sample 2 hours or 2 days after it has been created, because the microbial community will also change. So this is something that this needs to be taken into account very, very thoroughly. And we will be talking a little bit about this later in another session. But for any microbiome analysis, it is extremely important that all this is properly standardized and all these sources of biases are minimized. So I think we will chat a little bit more about this. But now sticking to this question, would any of you have any answer? What would be better for characterizing the interactions? So let's be more concrete, the biomolecular interactions between microbial species and the host.
Can you hear me?
Yes, I do.
Okay, so I think you are interested in the resident community of the microbiome. So I guess you would be interested in sampling like the intestinal tract.
Could you elaborate a little bit more?
Okay,
Why?
so I need to speak up. Sorry. Yeah, because I mean, I guess the interaction are going on. I mean, you are interested in the long-term interaction, something like that. So I suppose you're not really interested in the digesta itself, because it doesn't have any chance to interact, or at least it's a weak interaction, let's say.
Okay, so I would agree with you in most of the cases. So the microbial community that will be mostly interacting or directly interacting with the host will be the one that is closest to the mucosal barrier, to the intestinal wall, rather than in the digesta. Although for certain or many of the processes, the microbial community in the digesta will be also extremely relevant, because there might be some microbes that will be directly interacting with the epithelial barrier by producing some metabolites that then cells in the epithelial barrier of the intestine can detect. But there might be other bacteria also in the digesta that are digesting nutrients that later on will be used by other microbes in the intestinal wall that will be interacting with the host. So this is just an example that usually in this kind of studies, there is no black or white. There is a lot of gray zone, and there are a lot of direct interactions, and there will be also a lot of indirect interactions. And this is what makes this topic of research so complex, but so interesting at the same time. Something to consider here, and that we can observe from this figure, is that the diversity of the digesta is larger than that of the intestinal wall. So would any of you be able to explain why this could be? This is linked to what you mentioned about the resident community. If anyone else could develop more what a resident community is and how it's different from another type of community that you might already know. So perhaps the same person that answered this initial question, why do you think the digesta could be is more diverse than the intestinal wall?
Because I guess selection is acting. So, I mean, yeah, they are going through selection of the intestine and this kind of stuff. So, I mean, obviously the diversity is reduced because maybe I don't know, the condition in the tract of the intestine are not okay for developing this kind of stuff.
Mm-hmm. You mentioned selection, and we will be talking more about this in the second session, actually, of this. But now, what would you mean by selection? So, are you talking about natural selection at the evolutionary scale, or what kind of selection? What do you mean by selection? Would you be able to follow up on this?
Yeah, I think it's. Yeah, I mean, in the long term will be evolutionary selection, but I think it's more like a let's call an abiotic filter or something like that.
Mm-hmm. So you think that so there will be a bunch of microorganisms that will reach the intestine, but only let's say a subset of this will get attached to the intestinal wall. So that would be what you would be expecting. Okay.
Yeah, something like that. Yeah.
Yeah. In many cases, that's what will happen, actually. So there's kind of a filtering process because the microbes need to come from somewhere, and then some will be attached to the intestinal wall or very close to the intestinal wall, while some others will never be able to thrive in the mucosal barrier because their ecology is completely different. So there will be, in the case of marine organisms especially, or water living organisms, they will be filtering water all the time. So it is very probable that microorganisms that are in the water will also pass through the intestine. And these will be sampled in the digesta. And they can be sampled also in the intestinal wall. But the probability of capturing those compared to the microbes that are thriving in the intestine is different. So it is also important to think about this when interpreting the results of any of such studies. And linked to this, there is relevant questions. I added this as a group discussion because I wasn't sure what would be the arrangement. So I don't think we have any chance for a group discussion. So this should be an individual brainstorming, I guess. So would you have any thoughts on what does it mean to be associated with a host. When we say host-associated microbes, what does it mean to be associated? And this is related to the discussion we were having. Perhaps to make it a bit easier, I'd say, is every microbe we detect in the skin or the intestine of animals associated with a host? Do you have any thoughts on this? Yes, go ahead, please.
Yeah, I think that it's associated only if there is a construction or a destruction association. So an effect of these two directions, one of these two directions.
Okay.
In that case, it can be considered associated. So
Any other thoughts? What it means to be associated with a host? Yes, please.
It can also depend on the time. I mean, I imagine that some microbes can stay within an host for a certain amount of time, another for another amount of time. So there can be a kind of threshold to identify an association in respect to just a pass through.
Yep. Cool. Any other thoughts? Yes, please.
So it is. It means for me means that the host responds to the microbe by giving it by responding to its needs. Like at a time, anytime the needs of the microbe are met by the fact that the host is there and that there is an interaction between the two.
Yeah, so I would say so all these explanations are correct because there is no one explanation for what it means to be associated to a host. So we can think about occurrence, so perhaps just occurring within the intestine, we could consider this is to be associated with a host. There's a certain way to be associated. As it was mentioned, also there can be a time perspective, which is to occur for a long time. But there can be also a spatial aspect of it, which is to occur in multiple individuals or most of the individuals. We can also think about the capacity to reproduce inside the intestine, because there will be some bacteria that won't find a proper environment in order to reproduce. So perhaps we can define it in a way that these microbes that are able to proliferate within the intestine might be the ones that are associated. Or as it was mentioned, also could be the ones that influence somehow the host, that they produce some metabolites, some compounds that have an effect on the host, and the host reacts to this. So none of these answers is 100% correct, and the rest are 100% incorrect, depending on what are the aims, what is the system, what is the research question we have. The way that we will define the association can be different. So the important thing is to think really well about what we mean by the association. So in any research paper you might ever publish on this topic, just to very well define what you mean by associating to the host, because it can be very different depending on how we define that. I will add these slides, this kind of slides with interesting readings. We won't be able to deepen in most of the topics that we'll be covering. My aim will be to give you an overview of different topics, what I consider relevant in this research topic. And I have added some slides that if you have interest, then you can go to these publications and learn more about this. So you have an example here. Okay, so the next step will be to start exploring how animals and microorganisms interact at the molecular level. As it was mentioned before, there are some chemical reactions, there are some chemical compounds that either microbes or hosts can create, and this could affect the biology of the microbes or the host. So I have created this simplified scheme where we can start, for instance, from the microbes. We have the microbes here, and the microbes have these mumps. Have any of you heard about the mumps? What a mump is? For any of you that is working on microorganisms, have any clue? So mumps are molecular patterns that are associated with microorganisms. So MAMP comes from, it's an acronym of microorganism associated molecular pattern. And this is defined because we have two different types of patterns that hosts, animals in this case, can detect. The one is structural elements of the bacteria, in this case, or archaea, or fungi, so any kind of microorganism. And the other ones are the metabolites that microorganisms produce. So these MAMPs encompass both the components and the metabolites. Within the components, we can mention lipopolysaccharides, we can mention different kind of structural elements of the membrane, of the cell wall, of the capsule, the flagellum, etc., etc. While in the case of the metabolites, we might be talking about short-chain fatty acids, we might be talking about many other metabolites that microbes are able to produce. So these MAMPs will be identified by molecular receptors that animals have in their cells that are in contact with the microorganisms. These receptors are not only present in the intestine, are present in many different tissues, and their reaction could be very different depending on where they are and which microbes they get in contact with. But the important thing is that these receptors will trigger a response from the host, and this response can be in a way of producing antimicrobial peptides. It could be producing immunoglobulin A or other chemicals that will also influence the microbes. So we will not go in depth into these interactions, but this is just to let you know that there is this feedback, there is circular process where microbes affect hosts, and hosts have also the capacity to affect microbes biomolecularly. And this will be really challenging to analyze in a scientific context, because it is extremely difficult to establish causality. Whenever we are learning, we are studying animal microbiota interactions, we always will have the inherent problem of assessing the causality, because there are so many feedback processes, so many loops that will be affecting each other that it is very difficult to ascertain what is the starting point and what is the causal effect of this. So this is the direct response. It's showing the direct response that the hosts would be having on the microbes. But this response does not only happen directly with the cells that are in direct contact with microorganisms, for instance, in the intestinal wall, in the mucosa, in the epithelial layer, in the intestinal wall. These processes also trigger much larger systemic reactions. And this happens through different so-called axes that through hormones or different metabolites, multiple organs of the animal can be affected by these interactions with the hosts. We can talk about the gut-brain axis on how microbes might be affecting the nervous system and how changes triggered by the nervous system will end up also affecting the microbes. We can also talk about the gut-liver axis, because there will be a lot of compounds that animals produce, such as could be ketone bodies in the liver, that will be shaped or will be molded by the microbial community and their interaction with the animal. And this will also have an impact on the microbes. The same happens with the gut, and the same might happen with the lung. So there will be always these complex interactions that will have different effect sizes, of course, that will be affecting the host microbiota interactions in a systemic fashion. This is a figure of a simplified structure of a microorganism where we have the different components of the microbes. We have this rod-shaped bacteria that has the external capsule with a cell wall and a plasma membrane inside, with the nuclear DNA in the cytoplasm, and then a long flagellum and some pili in the capsule. So many of these elements will be MAMPs. Many of these elements can be identified by the host as elements that tell the host that a microbe, a specific microbe, is present in the close to the tissue. And these MAMPs include lipopolysaccharides, lipopeptides, lipoteichoic acids, flagellin, peptidoglycan, etc., etc. There are many ligands. There are many antigens in the end that will be triggering this response from the host perspective. It's not my aim you to learn about this, but this is just for you to know that there are many elements, many different MAMPs that will be identified by hosts. And this identification mainly happens through receptors, through cellular receptors that could be either in the membrane of the host cells, or could be also in the cytoplasm. So the most common receptors are toll-like receptors, TLRs, that appear in the cell wall of the intestines, of the host intestines. And these toll-like receptors will be able to identify some of these MAMPs and will trigger systemic responses from the host. There will be also some others, which are the node-like receptors. Those are in the cytoplasm, and this will be focused on detecting MAMPs that get into the cell. And this might happen because in some cases, for instance, macrophages, because the animal cells have phagocytated the bacteria, and the DNA that is taken out from the bacteria then can be identified, and that can trigger a different response. So the mechanistic process would be that we have these patterns, these microorganism-associated molecular patterns that are then identified by some receptors. These receptors are usually linked to an adapter, and this adapter is the one that triggers the response from the host. And then this response can be direct. This happens usually when the connection between the cell in which these receptors are and the bacteria are in close contact. These cells will be releasing immunoglobulin A, antimicrobial peptides. There are different cells that will be specialized in generating these different kind of molecules. But there will be also some other systemic processes that will be triggered that are much more complex, because we are talking about huge hormonal cascades that will be really changing a lot of processes in the host. So a small microbe can really trigger a huge response in the host. And mainly this will happen if it is a pathogenic bacteria, will trigger some kind of response from the host. If it's not a pathogenic bacteria, will trigger a different process. But the system or the underlying process is pretty much the same. There is also a huge bias, because all these studies on host microbiota interactions initially well focused on pathogenic interactions, on pathogenic bacteria interacting with hosts. Only in the last 10 years, let's say, researchers have really focused on what's going on with the rest of the bacteria that are not triggering pathogenic reactions or detrimental reactions for the host. So we still have this bias, and this will apply to many aspects of microbiome research, that the context at which these interactions were characterized will affect how the results were interpreted. So it is very important also to be aware of the context and to have a critical thinking on how to interpret the results, because most of the research, almost let's say over 90% of the research on microbiomes is done on animal-associated microbiomes is done in humans and mice. So that means that everything is extremely biased, and these biases need to be accounted for whenever we are working on mussels or fish or any other species that are not model species in which tons of data have been generated. So if you want to read more about all these interactions and processes, I have added 5 relevant papers here. So I will not deepen into this, but Do you have any questions about these interactions? Anything that you would like to know before we proceed? No questions? Okay, so now we will jump to another scale. So far, there's been interactions that happen at the molecular level between host and microorganisms. But also, I would also like to, well, before we jump to the evolutionary scale, I would like to ask you another question, which is, if you have ever thought how selective the response of the host can be over microbes. Do you think that the host reaction is the same for all hosts? And if it's not the same, this selectivity happens at the bacterial species level, bacterial strain level. What do you think? Have you ever thought about this? So who will dare to answer the first question? How selective do you think is the host response? Let's put it simpler. Is it species specific, bacteria specific, or is it general for all kind of bacteria?
They suggest species specific.
Okay, and how do you think?
Maybe because there's a coevolutionary mechanism in the interaction. They suggest specific.
Mm-hmm. So would you think then that there are different receptors for different microbes that have been in contact with the host? Or what do you think would be the mechanism to detect and to know each host to know which exact bacteria is in contact with them?
About the immune system.
Hormones.
Mm-hmm. It is true. There are some cases in which the host can be very specific in detecting some bacteria. There are some other responses that are much more general. So again, this is not black or white. There are some bacteria that can be identified at the species level. There are some others that cannot. There are some bacteria, for instance, that get completely coded by immunoglobulin A, while some others are not. There are some bacteria that produce very specific metabolites that hosts are able to detect, but some others produce very generic metabolites. So the response is that there is a huge variation, and that makes everything very complex as well. But now it's time for the next question, which is, what do you think are the implications of being more or less selective for the host? What do you think is best, to have one reaction per bacteria, or to have one reaction that will affect all the bacteria that get in contact or get associated with the host?
Somewhere between, I guess.
Trade-off. They suggest there is a trade-off.
There's a trade-off. Okay, can you elaborate a little bit more on this trade-off? Why is a trade-off?
No, it's better if you take out the mask because otherwise it's difficult. On, off, on, off.
Okay, I will try. I guess that being too selective entails that the host have to elaborate a lot of different reactions to organisms, to microbes. So it's a waste of energy, and the other way around, to not have any selective possibility to detect the bacteria. Do not permit to the host to take advantage of the positive interaction.
Mm-hmm. Yeah, no, perfect. So there is a trade-off, of course. So having all these mechanisms to detect different bumps in the air, it is costly. It requires to have all this machinery in place. And of course, the only way to have this machinery that is very microbe specific is to have some kind of evolutionary force that would first be mainly to keep any kind of capability that could have created during the evolutionary history of different hosts. So there are different levels of reaction, and depending on how the animals are able to react, the interactions, the evolutionary interactions they will have with the host will be very different. As I mentioned before, so there are some bacteria, for instance, that are affected differently by immunoglobulin A. And it has been shown that the level of heritability of this bacteria is different to others that interact in a different way with immunoglobulin A. So the level of vertical inheritance of bacteria will depend on how the hosts are able to first detect and then interact with this bacteria. So there are a lot of different examples, very different ways to interact, but the take-home message is that depending on how these interactions happen, in a very specific way or in a very general way, the evolutionary patterns between host and microbial interactions will be very different. So then the final part of this initial lecture will be on how animals and microorganisms interact, not at the biomolecular level, but at the evolutionary level. So what are your thoughts on this? So do you think that hosts and bacteria can co-evolve?
They say yes.
Yes. So those that think that they can co-evolve, could you try to define what co-evolution means? Can we say that, let's say, if there is a host with a phylogeny, well, let's go for a fish family, and we have different fish, and let's imagine this family contains 100 different species, and we will have a phylogeny. And then we have a bacteria that appears in all the different species, of the 100 species. If the phylogeny of the fish and the phylogeny of this bacteria are very, very similar, does this mean that they have co-evolved? What do you think? I don't know who said yes, but perhaps can be something that you can think about. Would you say that that's co-evolution?
What do you think? If the three match, is a co-evolution or not?
No, this description does not mention co-evolution.
They said no.
No. So then what is what you need to have co-evolution? So what I have described is a co-phylogeny, right? We have two phylogenies that are very similar, and they seem to be diversifying together. But does co-diversification equal co-evolution? Have you ever thought about that?
What do you think? Camille?
Evolution involves some interaction between the host and the bacteria, for example, and so it's like the red queen rule. One part of the duo has to evolve to get more, I don't know, maybe more advantage over the other one, and the other one is following along. They're not evolving in the same path because of the environment, but because of each other.
So would you say then that if a microbe and a host are co-evolving, they need to show some kind of functional interdependency, that they have to be affected. Yeah, they need to be, that they are in the end interdependent to prevent extinction of each other. Would be something like this.
Yes, that's what you
So this is what usually we would say. So co-phylogeny does not really meant to be, could be coevolution, or could be due to neutral processes that have not to be with evolution in the end. Because if there are no this kind of interaction, there will be selection over some traits in order to diversify, then we would usually not talk about coevolution. It is true that depending on how you define coevolution, this can be different. But I would say this is the most broadly accepted description or the definition, and that we should co-phylogeny should not be considered as coevolution. But there will be some cases in which coevolution happens, but that will usually not be the most common pattern, because mainly the temporal aspect and the biological features related to the temporal and the spatial aspects of microbes and hosts make very, very difficult coevolution to happen. There will be for sure co-phylogenies, but this might also be created through other processes. That could be some isolation of microbes, and they will be just diversifying, but not really co-evolving as an interdependency. There are some interesting reads about this. I will not deepen, but if you have any interest in this, I really recommend reading these 4 papers. So this is what I wanted to tell you in the first session. Do you have any questions, any thoughts you would like to share with the rest? No? Okay, so then we will have a 5-minute break now, and then we will start with the second session. Okay?
Okay, thank you, Anton. That's a 5-minute break. Thank you. Anton?
Yes, I'm here. Can
you hear me?
Yes.
Yes, I guess we can restart. What do you think?
Yeah, sure. Can you see
Okay.
the slides the way that you saw it before?
No, it's okay. It's okay. The slides are okay.
Super.
Okay, so please let's restart the second half of the lecture. Please sit down. Thank you. Let me start. So also the next day we have some colleagues speaking from their room and other colleagues online. So I suggest for the colleagues online to be organized with your own device and the headphone. And instead, if you want to intervene, make questions, maybe it's better if you speak up a bit and we use only this microphone instead of switch on off the volume, and because there is a lot of echo and interference. So please raise the hand, speak up straight into this microphone. It's much more interactive also, okay? So Anton, we can restart. And I have
Excellent.
a question for you.
Yeah, tell me.
What about the role of the immune system of the two players, the microbes and the host, in the co-evolutionary process? The immune system should have, I guess, an important role in this mechanism, in the matching or not of the two components. Please.
It does indeed, and I think it was in the slide 43 of the first session. You will find a lot of papers that deal with that. So it's mainly about the immunology and how the immune system has been shaped. Because there are a lot of papers, not just this. I have added some of the most relevant ones and some of the reviews that are available, but there are many original papers showing very cool stuff on how the immune system, and not just evolutionary aspects of the immune system, but also the developmental part, on how our own immune system is also matured due to the exposure of microbes to microbes. And I think that's one of the most important things, because as you know, we have different, so the immune system can be defined in different types. So that we have the innate one, we have the adaptive one, and we know that both are really affected by the contact with microbes. Of course, not just the adaptive one that is generated once you get in contact with an antigen, but also the innate one, how this is developed and how B cells, T cells are generated, etc. It is shaped on the interactions that happen between the host and the microorganisms in the early stage of the development. So it seems that it is a really critical period for hosts, the early stage, and how they interact. And this applies to humans and many other organisms. I haven't seen many papers on fish in that sense, but I'm pretty sure that this is going to be relevant as well. Most of the work is in mice and humans. But if you have any special interest on the immune system, there is a lot of literature about that. Okay, so the second session is on drivers and dynamics of animal microbiota interactions. It's based mainly on how these interactions change through space and time, and how this change. And this session will be somehow experimental, because I will be introducing you theoretical and framework that we are developing ourselves that has not been published yet. Will be published soon. I hope to submit this paper by the end of March. So this is the premiere of the framework. So I would like to see what is the reaction that you get. It is quite abstract, and it will require little bit of an attention. But if you are interested in these topics, I hope it will be interesting. And this mainly deals with factors that drive animal microbiota interactions. So if you were to list these factors, what would you list? Which are the factors that somehow shape animal microbiota interactions at the larger scale? I don't know if you can see this, but well, the small ones, but can you read what is written down in the thumbnail? No, okay, good.
No, I can't.
Otherwise, you regard the. So, any of you, would you like to respond? What are the factors? Just perhaps mention one factor that drive or shape animal microbiota interactions.
I guess the environment.
The environment. Yes, correct. The environment is extremely relevant on how these interactions will happen. Another element besides environment? Can you think of any factor that is not related to the environment that could have an impact? Yes.
They are models. They are not the presence of. No.
Stefano, can you echo that? I was unable to hear.
What can you repeat, please?
The abundance.
The abundance of
the abundance.
organism of the players.
Yes. Yes, it does. Anything else? Let's think about the host.
Time.
Is there any time? Of course it does as well.
The host.
But what intrinsic features of the host will also impact? Yes.
Can you repeat?
Social nutrients also.
Speak in the microphone, because otherwise I do the bridge, you know, like a radio.
Search of nutrients.
Search for nutrients. Yes, we can include that into the environment. So I will jump to the next slide, and I will just reveal what we have here. So I have just added these 3 points. Very simple. So all the answers that you have given are correct. But in this case, I will be mainly focused on environmental factors that we can call also extrinsic factors, microbial genetic factors, and host genetic factors. So whenever we are talking about these interactions, these 3 elements will be extremely important. And we'll be dealing with this across the next sessions. Then, of course, the interaction or the link that we have between the environment, microgenetics, and host genetics is not really even. We can also define this kind of fluxes of because the environment will be, of course, affecting the host and will be, of course, affecting the microbiome. And when a microbiome is in or on a host, there will be many factors that will be filtered through the host as well for the microbe. Right? So we cannot really think the environment as the external environment, but there will be many physiological factors, for instance, of the host that will be affected by the environment, but that will also define the environment for the host or for the microbes. So this makes everything a little bit more complicated, because from the microbial perspective, we can define the environment as in 2 layers. What could be the external environment, and the other one will be the internal environment within the host. And that external environment will directly affect the microbiome, for instance, by having a different diet, but could also impact the environment within the intestine by triggering these different kind of responses of thermoregulation, activity, etc., etc. So For this hopefully short lecture that I will give you now, I would like to focus on this. So how we define an environmental factor. I don't know if you were able to, I think you were able to read this one, so I will continue. So we will consider an environmental factor any factor that is not encoded in the genetic material of Microsoft hosts. You can define environmental factor in very different ways, but in this case, we will define it this way. So it's just to set a dichotomy between the genetics and which is intrinsic to a host and the rest of the factors which are not intrinsic to the genetics of a host. So diet, for instance, will be an environmental factor, temperature, humidity, habitat, interaction with other hosts, etc., etc. All this will be defined as environmental factors, while genetic content of the host will be considered a genetic factor in the end. So whenever we are talking about environmental factors, we'll be talking about, as I said, diet, temperature, humidity, which are the environment, the outer environmental factors. But it's going to be also very important to pay attention on the developmental stage, physiological state of the host, which will be also affected by the environment. But having these two scales, or the two different layers of environmental variation for the microbes is going to be very important how we model, in the end, the interactions between host and microorganisms. There are many papers on this dichotomy about the genetic and environmental factors. I have added a couple of them. I could also add some more, but these two would be a good starting point if you have any interest in reading more about how genetics and environmental factors, or intrinsic and extrinsic factors, affect host microbiota interactions. So now going to the point, this session, as I said, it's going to be a little bit of an experiment with you, because I will introduce for the first time to a let's say, specialized audience, the framework we are building on the metagenomic space concept. So this is the framework we are building. It's going to be a theoretical, but also a mathematical framework in order to study animal microbiota interactions. I will try to explain you what are the foundations of this metagenomic space concept and the surrounding concepts that will be relevant in this framework, and also just some highlights of what is the relevance of this, or what we believe it is the relevance. So I have this GIF as well, which is, I think it will require some special level of attention, because there will be quite a lot of concepts that I will introduce now, and I hope I will manage to explain it properly so that you can follow. In the end, it's not that complicated, but it is very conceptual in the beginning, at least. So let's hope it is not too painful for you. So this framework starts by defining a metagenomic state. And we can define a metagenomic state as I think I added the actual, yeah. So we can define a metagenomic state as the genetic conformation that a microbial community associated with a host exhibits at any given time point. So a metagenomic state will be at any host and at any given moment, what is the genetic information that the microbial community carries. This can be defined in different ways. So in the case of the A figure, you can see the actual microbes. You can see that there are 5 green microbes, 3 yellow microbes, 2 red or orange microbes, and 1 blue microbe. So this could be characterized by the relative abundances, for instance. And we can just abridge or reduce all the information about the genetics that we could potentially have to relative abundances of the taxa of interest. So not really paying attention to what are the genetic, so the gene contents that each of these taxa contains. I'm quite sure that Marco in the morning explained you the difference between targeted amplicon sequencing approaches, the 16S, and shotgun metagenomics. I guess he said this difference. So in the case of this figure B, this would be what we would get by analyzing 16S amplicon sequencing data, for instance. We will know which O2Us or which ASVs, which microbial species will be there, and we will know what are the relative abundances. So this will be the way to define a metagenomic state. But if we use shotgun metagenomics, in which we reconstruct the bacterial genomes, then we can define a metagenomic state as at a much finer resolution. And we can do that by defining which functions are present, for instance, in the microbiome in that metagenomic state. And if we even complicate this more and we include metatranscriptomic information, for instance, gene expression, we can also characterize a metagenomic state based on how microbial genes are being expressed and what are the relative expression values. So these are different ways of defining the same concept with different levels of resolution. But the idea is that this metagenomic state will be the unit that we will be using for creating or building up more complex properties and features. Because metagenomic states change across time. So here you can see different examples on how we can characterize the change of the metagenomic states from the initial one to the two different ones. You can see in the figure E that the relative abundances of the different microbes will change, and this can be shown in figure F as the changes in the actual relative numbers of bacteria that we have in the community. But then in G and H, we can show the functional differences that have happened. The take-home message here is that metagenomic state is the unit that will be used for characterizing the microbiome. And these metagenomic states will change through time and will change also across different hosts. So based on this metagenomic state, we can try build a metagenomic space. We call a metagenomic space, which is the total possible array of different metagenomic states that the host could exhibit. So a metagenomic space is any kind of microbial arrangement that a given host can exhibit, could potentially exhibit, with important information there in the parentheses that does not necessarily need to exhibit. But this is a theoretical property of a host. So could you think of any kind of relevance of the metagenomic space? Why this can be relevant?
Depends on the field of application, but I think that it can be useful from defining a diet for animals. I don't know who do you call I don't know, farmed animals. So it's a way to understand which possibility what you can try with the diet of this animal. So what is the best for its life? And for a total different perspective, you can find what study related to ecological niche of wild animals also, especially for animals which habitat is threatened and it's redundant.
Mm-hmm. Yes, so at the applied perspective, once we have a specific metagenomic space, this can have an impact on the biology of the host. And this is one of the objectives of our framework. Because in the end, each of these metagenomic states can provide either benefits or some kind of detrimental effect on the host. The entire space that the microbial communities might take within a host will define what is the potential contribution the microbiome can do to a host. So any metagenomic state that is beyond the space of a given host, it's something that will never be possible to happen. While everything that is within the metagenomic space is a microbial community that might occur within a host. So that defines the difference between what can be the contribution the microbes can do to a host, could be beneficial or detrimental, compared to what microbes cannot do. And this can be done by modeling the metagenomic space. So if we take this example in which we have characterized 10 different metagenomic states, you see the cells that are numbered here are the ones that we have sampled, let's say. This is all a theoretical representation. But let's imagine we have 10 points in time from a specific animal that have been sampled. And each of these metagenomic states is a different state. It's a different arrangement of the community. But there will be also some stages that we are unable to capture because, of course, we cannot sample the animal all the time. We cannot really know what is the exact metagenomic state of each microbiome at any given moment. So we will need to somehow mathematically model what is this space that we can take. And this can be done mathematically. It's not that complicated, but visually we'll be representing something like this, in which we have different metagenomic states. Each of these hexagons is a different arrangement of the microbial community or a different pattern, gene expression pattern. And the color will show what is the probability of this metagenomic state to occur within a host. Is that more or less clear? This would be something, if you have ever done some ecological niche modeling or something to try map in space, what is the probability of occurrence of a species? This would be something similar. So in the end, we are talking about what is the probability of finding a microbial community within the host. I will try to explain you why this is relevant in the end of this lesson. So, as I mentioned earlier, the metagenomic state is dynamic. So the microbial community changes all the time within the hosts. That's something that happens, but these changes can be very different. And our framework is based on, it's focused on trying to discern different types of changes depending on how this impacts on the host. So here you have a representation in which you have one axis of the metagenomic space. Or in this case, we have two axes. But this is meant to be a simplified representation of a multi-axis volume. So when we do the actual mathematical modeling, we will not be reducing this to two axes. This can have as many axes as we want in order to create a hypervolume. In this case, for the sake of a simplified representation, we will be just showing one axis of the microbiome change. And this axis might be, we can define one axis, for instance, could be, if we think in a functional way, what is the capacity for producing short-chain fatty acids, for instance? Let's imagine that's the only axis we are considering on how we are characterizing the microbiome. Another type of one single axis could be what is the proportion of the anaerobic bacteria. It could be also what is the proportion of gram negatives, etc., etc. So there will be many different properties that we can use in order to characterize the microbiome. So in this representation, we will just be using one of them in order to simplify what I want to show you. So here you can see the metagenomic states. Each of these hexagons is a different metagenomic state. And the oscillation of these metagenomic states defines the potential metagenomic space. We have here the metagenomic states, and that defines the potential metagenomic space. And we define the potential metagenomic space as the array of metagenomic states that a host can exhibit in the absence of measured variation of external influences. So in the human, mainly in the human microbiome literature, this has been defined as the dynamic equilibrium. So when the environment doesn't change, when the physiological features of the host doesn't change, the microbiome changes. The microbiome has its own dynamics because we are not talking about one element, we are talking about thousands of elements, a community of living organisms that are interacting with each other. So this is something that we will be observing all the time. Even if anything else doesn't change, apparently, anytime we sample a microbiome, we will be getting something slightly different. Of course, there will be some technical biases and distortions, but even biologically, there will be virtually no chance to have exactly the same microbiome in 2 time points within the same individual, even if the environmental factors have not changed. So this is what we define as the potential metagenomic spaces, how this drift of the microbiome happens in the absence of environmental changes. Is this more or less clear?
Yeah.
So some animals will show a larger metagenomic drift than others, but there might be some animals that have a very minimal oscillation, but there will be some other animals that have a larger oscillation. And this will have a different, we will define this as a different metagenomic drift. So the potential metagenomic space of each individual will be also defined by that. So, as I said, that happens when environmental conditions do not change. But what happens when the environmental conditions change? We have 2 main options. One is that the potential metagenomic space will not change. That is that the microbiome does not react to the environmental change. There will be some environmental changes that will not have an effect on the microbiome. We can think of, for instance, the moon phase. Perhaps it does in some cases, but for most of the animal-associated microorganisms, the effect of the moon might not really be relevant enough to change the condition. But perhaps for some plant-associated microbes, it is. So there will be some conditions or environmental changes that we might define that can trigger a change or not of the potential metagenomic space. This is the example in which there is no change. We change the environmental conditions, but everything continues the same. We observe no change. But there's also this possibility in which when we have a different condition, the microbial community reaches a different stability. And we call this the metagenomic space transition. We transition from an initial dynamic equilibrium state to a new one. And defining these properties will be relevant for modeling this mathematically and then to think about how these host microbiota interactions can change and what is the effect they have on the evolution of microbes and the hosts. So far, I first introduced the metagenomic state concept, which was each of these microbial community arrangements at any given point. And the second concept that was introduced was the potential metagenomic space, which is how these metagenomic states change in the absence of external influences, environmental changes. And the third relevant one is the fundamental metagenomic space, which is the array of metagenomic states that a particular host could conceivably exhibit, considering all possible combinations of biological states of the hosts, environmental conditions, accessibility of microbes, etc., etc. So the main difference between the fundamental and the potential metagenomic spaces is that the potential is defined in the absence of environmental variation or the host physiological variation, while the fundamental is defined as the general one, the absolute maximum of metagenomic states that a host can exhibit at any given point. So this is a representation of that. Here in figure A, we can see that there was an initial condition where we had a potential metagenomic state in the beginning. This changed. There was a transition after the condition 2 happened. There was a new transition when the hosts were subject to a different condition. And then when returning to the initial condition, the potential metagenomic space returned to the initial phase. So you can see that all this variation would be defined in the fundamental metagenomic space. if this is if this would be sampling theoretically the entire possibility of different environments possible. And we can also show that in a two-dimensional space where we have the potential metagenomic space here in yellowish colors. Then it jumps to a different one, which is the greenish colors, and then it jumps to the bluish colors to return again to the yellowish colors. And all this area that is encompassing all these metagenomic states and spaces will be defined in the fundamental metagenomic space. And this can be, of course, modeled the same way as we did in the beginning. The fundamental metagenomic space will be defined by would be a mathematical process to see what is the probability of showing different metagenomic states. There are many other concepts that can be defined. The metagenomic plasticity is defined as the capacity to jump from one potential metagenomic state to another, while the metagenomic evolvability would be defined as the capacity to change the fundamental metagenomic space through the evolution. So all these concepts will be defined by different factors, and this will have huge impacts on how we analyze animal microbiota interactions. I will not deepen too much into this, because I know it is quite abstract. We will reach a point in which I will show you some more specific example in which we will try to make some sense to all this conceptualization. But the main question that we need to ask ourselves is, which are the factors that do determine these metagenomic spaces, both the potential and the fundamental? So within our framework, the fundamental metagenomic space is determined by intrinsic host genetic factors. That's how we conceptualize all this. And there are some benefits in doing this, because we would be defining a range of microbial communities that a host could exhibit only based on its host genetic factors. And these host genetic factors includes variation that ranges from very micro scale microgenetic variation that could be variations in the immune system, to variations that are much more conspicuous, that could be on the morphology of the intestine, for instance. This means that, for instance, ourselves, humans, will never be able to exhibit a metagenomic state that ruminants are showing in the rumen, for instance, because the physiological conditions, the morphological conditions are completely different, and it will never enable having an identical microbial community because of these fundamental changes that exist. While the potential metagenomic space is considerably more complex, because it will be determined by combinations of environmental conditions, but also host physiological conditions. The most relevant thing is that all these concepts, that could be the fundamental metagenomic space, potential metagenomic space, metagenomic plasticity, metagenomic evolvability, are measuring different aspects of these host microbiota relationships. And these have not been explicitly characterized or defined and characterized in the literature so far. So the reason why we have done this exercise of creating these concepts and organizing them within a meaningful framework is because we feel that there are many features of the microbiome that are analyzed using different approaches, and sometimes what researchers are analyzing is not really what they think they are analyzing. And I will try to explain that the following. So let's imagine here that we have two hosts, and you can see that the first plane, that one that is on the top, shows the metagenomic space of the first host, while the second one is the second host. So would you say that these two hosts some similar or different metagenomic spaces?
Similar or different? Similar.
They are similar, right? They are very, very similar. And this can be, the importance of this is that if we take these three vertical polygons here, the blue one, the yellow one, and the pink one, This is showing the same metagenomic state. And you can see that the colors are pretty much similar, which means that the probability of detecting each of these metagenomic states in the two hosts is pretty much similar. However, if we have this option, we have two hosts again, but the metagenomic states are different. So metagenomic spaces are different. We may be talking about the potential or the fundamental, but here the main thing is that the probabilities of having metagenomic states is different. So for instance, in the case of the yellow polygon, you can see that there is a high probability of finding this metagenomic state in host 1 because it is purple-ish, while in the other one, in the second host, it is oranges, which is a lower probability. Most relevantly, in the case of the blue polygon, there's a small probability of finding this metagenomic state in host 1, but that metagenomic state is beyond the space of the second one, which means that this will not be found in the host 2. And this is a big difference, because it means that this metagenomic state will never be able to do anything to that host because it is impossible to be arranged within the host. So our framework is mainly developed to try make sense to this kind of small differences. But I will try to explain this by a concrete example. I don't know if you are familiar with the microbiome literature, but some of the most well-cited papers are those by Woodrich and colleagues on twin studies. So this researcher and her colleagues did two big studies by comparing monozygotic twins against dizygotic twins. And they used this to show that there is some signal of the genetics on how the microbiome differs across hosts. But they focused it in a way that let's contrast whether it is environment or the host genetics, or it is affecting the microbiome variation across hosts. Individuals, in this case, human individuals. And the approach they used for this was to compare monozygotic twins against dizygotic twins. The issue we have here is that what we should be expecting, as monozygotic twins are genetically identical, we should be expecting that the metagenomic space, the fundamental metagenomic space, to be exactly identical. So the probability for detecting each metagenomic state, in principle, should be identical. Of course, then this will be modified by the environment within the space. But if we talk just about the genetics, then it should be identical. In the case of the dizygotic twins, metagenomic spaces are slightly different, but only slightly different. So it is very improbable to find metagenomic states probability differences on the differences on the probability of finding the same metagenomic state. This means that if we sample this in, so let's imagine that these are two twins, for instance. So the possibility of finding this, what we can observe in this blue polygon, is extremely improbable. And this would be the only way in order to find a genetic signal, which are differences in the probabilities of finding different metagenomic states. I know this is a little bit complex. I will try to use another example that perhaps makes more sense. Let's imagine that we have two species now here. And you can see that the species 1, with its potential, so with its metagenomic space in the higher, in the top plane, you can see this how the wild population is determined by this fundamental metagenomic space. And you can see that the species 2 that is on the bottom, that has a completely different metagenomic space. But there's a very small overlap. These two. Can you see this small overlap between the two planes? So let's imagine that the only way that you can find this overlap is by putting these two species in the same tank under the same conditions for many, many weeks, let's say. But in the end, the environmental conditions will be so identical that both the genetic effects that are teasing this apart, these two microbiomes apart, will be getting closer and closer and closer, and there will be just a very small chance, but a real one, to show the same microbiome community. Let's imagine that we use that microbiome community, as a characteristic microbiome of that species. Would you say that that is a good way to characterize the microbiome of this species? In the case of a species 1, we have this blue polygon that defines what is the, let's say, the most probable metagenomic states that we can find in this species. In the case of the species 2, it is the yellow polygon, which is showing that. And we will be sampling what is in the pink polygon. Would you say that that is characteristic of this species? It is certainly within the fundamental space of both species. But I could see that there was a hand that was about to be raised. So please, go ahead.
Maybe it's not characteristic of the species, but more of the active condition.
Mm-hmm. Yes, that's true. So it is within the space that species have, but it is in the very edge of it. And this is probably what we are observing, for instance, whenever we use zoo animals for characterizing the microbiome of wild animals. So let's imagine that the same example, in this case, I put fish here because we were talking about here, we are maybe in a marine setup. But let's imagine that we sample a lion in the Chicago Zoo. Would you be expecting that the microbiome of a lion in the Chicago Zoo will be representative of the microbiome of lions in the Serengeti Savannah? So this will be something similar. It is important to know what we are really characterizing. And when we compare a species, we should try identify first what is the most probable metagenomic state that is the characteristic one of that species. Because the inferences that we would be doing from getting different kind of information, either the one that is characterizing the center of this metagenomic space or the edge, might have completely different results when we compare the species. If we sample the orange or the pink part, both species are very, very alike in terms of microbiome. If we sample the centers of their metagenomic spaces, we will be concluding that they are very different each other. But this is one of the examples why this framework might be relevant. This is the same example here shown in a different way, just on a single axis. In the picture B, you can see the density curves of the metagenomic states of species 1 and species 2. You can see that the one that we would be sampling, it is not representative at all, either two species. It is within the space, but it is in the very edge. So it is important to know what we are sampling in order to interpret that properly. So you can see the example. So this was the first attempt to introduce you the microbiome, so the metagenomic space concept, which has huge implications, mainly in the evolutionary aspects of this, of the host microbiota interactions. I will not deepen more into this. I have another set of slides on this, but we will run out of time, I'm afraid. So I think unless you have a special interest in following up with this, I would be stopping with this. Do you have any questions you would like to address at this point? I know it is quite abstract, but perhaps for that reason, it would be, yes?
Is this spatial metagenomic useful also to forecast the effects of some changes on the microbiome of some species?
Some changes of what, sorry?
On the microbiome of some species, so their health maybe or their conditions, It's a tool to forecast.
Also, yes.
It is mainly so. The microbiome, it depends on how we define them, but it is once we manage to model these metadynamic spaces, then we should be able to have some probabilistic values of having some specific arrangement of the microbiota. Depending on what we are modeling, whether it is the fundamental metagenomic space, the potential metagenomic space, or it is the metagenomic plasticity, etc., will be different ways of analyzing this. But it is indeed a way that could enable us to model and to predict how the microbiome might change in different hosts. Our main focus is the evolutionary one, but it is also relevant in the applied aspect. So one example would be, for instance, when we compare the effect of environment versus host genetics, for instance. So let's imagine that we have a certain degree of genetic dissimilarity between hosts, and that yields a slightly different microbiome, metagenomic spaces. So it could be something similar to this. So we can have multiple. I will use the chicken example, for instance, because it's something that I will work with chickens. So farm chickens are extremely alike genetically. They are very, very similar genetically. So compared to the wild chickens, for instance. However, so environmental changes that happen then would have a much larger impact than the genetics on the microbiome. That's something that we should be expecting. When we have hosts that are genetically very alike, the environment will have a much higher impact. However, how much the environment will change will determine how relevant the genetic variation will be. Because if we focus on farm chickens are raised in very environmentally constant conditions. And we compare those to wild chickens will be experiencing very different environmental conditions. In the case, if we observe the same level of genetic variation, depending on how large the environmental variation is, the effect will be completely different. Because if we have the slightest difference in the genetics in a farm environment with where the environment is very constant, the effect of the genetics will be raised and will be really surfaced. While in a wild environment, this would not be surfaced. So different changes in the metagenomic space will have different impacts depending on what is the context itself. And we will focus on that in the last sections, in the last session of this teaching session later. So our main focus is evolution, but it has also impact on the applied aspect. Great. But if you don't have any other questions, let's see what's the time now. And the next session is completely different. This was somehow experimental, and I'm sorry if it was too conceptual, but the next one will be quite different. So we are 10 minutes behind the schedule. So what we can do is now start with the next session, and I will see whether we can stop at some point. Is that okay? Do you have the energy to go for the next session, or would you prefer to have a short break? Stefano, what's your?
I'm trying to understand from the student. Would you like to go on or want a break? Move on.
Let's go for that then. Let me open the next session. Wait, can you see the slides again? So now, after this somehow experimental session, now I will jump to the core of this lesson, which is the hologenomics. And I would like to ask you one thing, first of all, just have you ever heard about hologenomics, and would you be able to define it somehow? Would any of you dare to define hologenomics? Do you know what it is? Has any of you ever heard it before? Can you raise your hands, the ones that have heard about this before? Okay, cool. So we are facing something that's completely new then. Now, this is the definition of hologenomics we included in this paper that we published in Nature Review Genetics. And this will be one of the main central points in this following two lectures. So we define hologenomics as the integrated study of the genetic features of a eukaryotic host alongside that of its associated microbes. So it is basically to do genetics or genomics and metagenomics at the same time. Usually focusing something that relates to both parts of the system, the microbial and the host part. And in order to define hologenomics, of course, we need to define the hologenome. And what the hologenome is the entirety of the genetic information contained by the eukaryotic host and the microbial community associated with that host. So this is how we will be defining the hologenome. And we recently published this paper that I mentioned before on disentangling the host microbiota complexity through hologenomics and how we can do that using not just genomic and metagenomic methods together, but also different kind of multi-omic methods in order to understand how animals and microorganisms interact. So in order to start discussing and talking about hologenomics, I think it is important to look back and see how we have so far tackled these animal microbiota interactions. Well, no, in this case would be how we have historically tackled how the phenotypes of animals, in this case, is affected by different kind of factors. So historically, we have been mainly working on this left option, which is the genomic approach. We have tried to predict phenotypes based on the genomic, the interaction between the genomes of the hosts and the environment. More recently, researchers focused on the microbiome and tried to predict phenotypes based on the microbiome or the metagenome interacting with the environment. What we are saying with hologenomics is that neither the genomic approach or the metagenomic approach is complete enough to show the whole picture. So what we need to do is to use the hologenomic approach, which is to find interactions between the genome, the metagenome, and the environment, and see and try to model how these interactions can predict certain phenotypic traits in the hosts. This is just the very simplified equations that we will be using. So this is trying to model the phenotype by equating the genome with environment, trying to model the phenotype equating the metagenome with environment, and then using the three together, and mainly focusing on the interactions. So in order to do that, what we do in our research group is to generate multi-omic methods, more multi-omic data for both the host part and the microbiome part, in order to try disentangle what are the molecular interactions that happen between one and the other. Sometimes just generating genomic data from the host and metagenomic data from the microbiome is enough. In some other cases, and depending on what are the research questions, we'll be focusing more and more and generate gene expression data, proteomic data, and metabolomic data, which could be used for validating different observations that we do. So in the following, I will give you an overview of what is the relevance of having different, making different choices along the process of generating and interpreting this data. And this starts from the collection and the preservation and the destruction of the nucleic acids of the proteins from the samples. So this will be somehow linked also to one of the questions that came across the beginning of this session. But one of the first things that we will need to take into account is that in order to do hologenomics, of course, we need to generate data from the host genome and the microbial metagenome. The simplest way to do this would be to try characterize the microbial metagenome by extracting and analyzing the DNA from the microbial community, and do the same from the host by collecting blood samples or tissue samples where we can have microbe-free DNA that enable us to generate data on the genomic features of the host. However, there are many things that we need to consider when we do that. And one of these is the biases that we can have when we extract the DNA from the microbes, for instance. Here you have two examples of different cell walls of bacteria, comparing gram negatives against gram positives. Now, the thickness of the cell wall of this bacteria is not identical. So the probability of these cells to be lysed when we do the DNA extraction will be different. So this is something that we need to take into consideration whenever we do any kind of analysis of DNA analysis. And this does not only apply to hologenomics, it applies to any molecular analysis we do on microorganisms and their communities. This is an example of a paper which Hale and colleagues showed that depending on what is the preservation method, the amount of DNA you use, microbial DNA you extract, and the microbial community you recover is different. This is not based on marine organisms, but it would apply equally well to marine organisms, because it is extremely important to consider which DNA sample preservation and DNA extraction methods we'll be using. And this is spatially relevant on the microbiome. This is not that relevant to characterize host genomes, but it is extremely relevant to characterize microbial metagenomes. And this same publication, we could see these changes where the two bars, stacked bar plots that you can see in the left are fresh samples that we should be expecting what would be closest to the reality. And then you can see, depending on what is the preservation method you're using, you will be able to recover different microbial communities, some being more alike to the original than others. Of course, using the sampling methodologies and preservation methodologies that keep the microbiome community as similar as the original is the best, but it's something that cannot always be achieved. So if you were sampling in the field where you don't have access to dry ice, or you don't have access to liquid nitrogen, is this extremely challenging to snap freeze the samples and keep them frozen until these are transported to the lab. So there are different options for this, but the take-home message would be that even though some methods are better than others, the key point here is always to keep consistency. Use always the same preservation and DNA extraction methods in order to generate comparable data. And this is extremely, extremely important for microbiome research, because in order to compare microbial community data, we need to rely on the same processes, because otherwise the comparability drops considerably. And this is one of the main challenges we are facing nowadays, that different groups use different approaches, and it is extremely difficult to compare data generated by different groups and different studies because they don't follow standardized methodologies. I think that Marco showed you this morning the example of the Earth Microbiome Project. So we have a similar project that does not only focus on generating information of microbial communities, but also the genetic properties of the hosts that host these microbial communities. And it's called the Earth Hologenome Initiative. If you have any interest, you have the link to the website here. And what we do in this project is to generate and analyze the jointly host genomic and microbial metagenomic data. In this case, we send to our collaborators this sampling kit that contains tubes that are pre-filled with some specific buffer. And this buffer is different to the one that is just in the Earth microbiome project. And the main reason is that we had some ethical constraints that we set to ourselves, which is that we think that we cannot standardize methods that rely on commercial solutions. So one of the main caveats or limitations of the Earth microbiome project, from my own perspective, is that they standardize a extraction kit of a commercial provider, which to me is an extremely bad idea. One, from an ethical perspective, because you are standardizing something that clearly benefits one company. And the other one is if you become completely dependent on that company. If that company stops generating that kits, then you are done. So I know that the easiest way is always to rely on commercial kits, but if you ever gonna run a very long-term study, it might be a good idea to rely on homemade solutions. That would be my advice, at least, because you can never know when the production of a DNA extraction kit or a library building kit, etc., will be stopped to be produced. So this is something that we are trying to avoid in the Earth Hologenomics initiative by using open access methods that we have developed in our lab. They are completely open access, so no commercial limitations. And this can ensure that anyone can get access to these procedures. This is just something that I consider is relevant to keep in mind whenever you are doing molecular work, and mainly when you are working with microbiomes, because it is extremely sensitive on the methodologies you use in the lab, the characterization of it. So one question that we will need to ask ourselves whenever we generate microbiome data is how much sequencing we need. And this is important to keep in mind because a microbial community is a complex community with usually with hundreds and sometimes with thousands of different microorganisms. So the genome size of these microorganisms is usually between 2 and 7 megabases. So this is between 2 and 7 million base pairs of the genome. Now we need to keep in mind that, and if we use the current sequencing standards, I don't know how much you know about sequencing platforms, but this Illumina NovaSeq is one of the largest platforms that yields most of the data, and it is quite heavily used nowadays. So one lane of an Illumina NovaSeq S4 run yields 800 gigabases of data. So this is the same as 800,000 megabases of data. So this would mean that one sequencing run would be able to sequence a lactobacillus genome 400,000 times, an Enterococcus genome 260,000 times, etc., etc. So nowadays, the sequencing capacity we have exceeds by far what is the genome size of a single bacterium. And this is why what we do is when we do the sequencing, I think that Marco mentioned this in the morning, we don't generate one piece of data for each sequencing run. But what we do is to multiplex. When we build the libraries, we use different indices so that then bioinformatically we can pull multiple samples together, generate the data all together, and then bioinformatically retrieve back which sequencing reads belong to which samples. So in our case, in our personal case, for instance, in our lab, we are able to multiplex 288 libraries together in one lane. So we can generate sequencing data of 288 samples at the same time. That means that rather than generating 800 gigabases of data per sample, we might be targeting about 8 gigabases of data, more or less. And this still equates like 4,000 times a lactobacillus genome, 2,600 times an Enterococcus genome, etc., etc. Now, what we need to keep in mind is that the microorganisms appear in very different relative abundances. There is a possibility that we might have 100 times or even 1,000 times more abundant bacteria compared to others. So in order to characterize this blue bacteria properly, we will need to sequence much more than what is the genome size of this bacteria. And that is why we generate a lot of redundant data. The more even the microbial community is, the relative abundances of the microbial community is, we will need less sequencing. The more uneven it is, we will need more sequencing. The less diverse the microbial community is, we will need less sequencer. The more diverse it is, we will require more sequencing. So, and finding this balance, it's not always easy. That's something that you need experience, and it is quite challenging to achieve because there are many things that need to be considered, and even more when we do hologenomics. And this is because there is one important thing that whenever we analyze host-associated samples, is that the samples will always contain a fraction that belongs to the host, a fraction that belongs to bacteria, and then a fraction that belongs to other taxa. If we are interested in doing just microbial metagenomics, we want to reduce as much as possible the host fraction and the other fraction, right? We want to have as much bacterial DNA as possible, because otherwise all the sequencing data we generate will be just what usually has been called as contamination. So it is kind of useless sequencing, and sequencing is expensive, and processing sequencing data is expensive. So we will try to minimize that. When we do hologenomics, though, we are also interested in generating host data. So it is quite interesting to have both host DNA and bacterial DNA in the same sample. So doing hologenomics is also a way to make the most of the data that is contaminated with host DNA. So going back to this initial slide where I showed that getting microbial DNA and host DNA, sometimes it's not necessary to get pure tissue samples of fish in order to characterize the genome of the fish, because the microbial, what we were targeting, the microbial DNA, will contain enough host DNA in order to characterize the host as well. So running some preliminary analysis on what is the host microbiome fraction is quite interesting, because that can save us a lot of time and money. These are three different options we have that we will also find quite often. So we might have different fractions of host, bacterial, and other DNA. And depending on how this is related to each other, so what are the relative fractions, the amount of sequencing we will require will be different. This is an example with chicken data, for instance. So in a European project that we are running nowadays, we have generated data from two intestinal sections of the chickens, the ileum and the cecum, and the data that we retrieve is strikingly different. So in the case of the ileum, we have 95% of host DNA, and in the case of the cecum, we have 95% of microbial DNA, and both are digester samples. Both are intestinal content samples that have been extracted from the intestinal, from the gut of the animals. So depending what we have here, the approach that we can use for generating microbial metagenomic and animal genomic data can be very different. For instance, if we were relying just on the ileum, as 95% of the DNA is host, we won't need to generate more host DNA from a pure sample, because any data we generate aiming microbiome will yield us a lot of host DNA. And this is what we often observe in fish. If I show you this, we go back to this example, we also did shotgun metagenomics on this same fish that I showed you before. And what we observed is that in the intestinal wall community, if we do shotgun metagenomics, over 99% of the DNA belongs to the host. While if we do the same in the digester community, about 90% belongs to the host. And even though these two might seem to be a small difference, it is a big difference in the end. Because in order to generate enough data for characterizing the microbial community of the intestinal wall, we will need to generate tons and tons and tons of host DNA as well. And if we are not interested in generating host DNA, then it's going to be an extreme investment in sequencing to only use less than 1% of the sequencing data that we produce. So this is something very, very relevant to keep in mind whenever you start designing studies that are based on shotgun metagenomics. And this is especially relevant on fish and marine organisms because they are usually, the bacterial fraction, it is very often very, very low, which complicates using shotgun metagenomic approaches. This does not affect that much amplicon sequencing approaches like 16S rRNA gene amplification, because even if we have a very small fraction, that fraction will be amplified and will be characterized. But when we do shotgun metagenomics, we rely on total DNA, and it is very, very challenging to do this. What we do in our group is usually to use hybrid methods. We combine 16S amplicon sequencing with shotgun metagenomics. But you need to be aware that very often it is impossible to get deep functional inferences from this kind of data because you will end up with very little amount of data that is of relevance for characterizing the microbiome. So if you ever plan to do this in the future, this is something you should really consider because it is very, very important from the practical perspective. And of course, then there is the intrinsic difference because the only way to get shotgun data from these animals was to characterize the digesta. From the intestinal wall community, we cannot do really anything. But from the digesta, of course, you will be characterizing a lot of transient microbes and not the resident ones. So you will always need to find this trade-off between what you characterize, which taxa you characterize, which ones are characterized functionally, deeply, etc., etc. It's quite complicated and really worth a thought before designing the projects. So the next step will be on bioinformatics, and I will go through something that's I think that's Marco has covered in the morning a little bit. But before entering into this, I think it would be nice to have a break. Would you like? Do you have any questions on what I have introduced so far on the sampling, preservation, DNA extraction, etc., etc.?
Question on the methods? Yes, one.
Yeah, I have a
Yes,
Thank you.
About the plot you showed us with the concentration of DNA depending on the way we fix it. And the question is why, regarding the freezing methods, the content seems to gets higher in levels. Or maybe I am a misunderstanding, I don't know.
Yeah, so I haven't really checked this. I don't know why. So these are, they characterize the sample when they sample it, and then they preserve this in 5 different preservation methods for 1 week, for 2 weeks, for 4 weeks, and 8 weeks. What you are asking is why the concentration is much lower in the first week, right?
Yes, in particular for the treated ones. Yes.
The minus 80, you mean?
Sorry?
The minus 80?
Yes, minus 80 or minus 20.
Why the frozen, the minus 20, minus 80 samples are so low in the first week?
To be honest, I don't know. We will need to go back to the article and see what's they might be, what might be the explanation to this, because
For me, it's okay.
No, I think I don't really know, and I don't know if it has to do with the way that the samples were structured, or the actual freezing might have any kind of impact on the. To be honest, I don't know. Usually, I have never run myself this kind of tests at using these methods. I have done it using our preservation methods. We are using the Earth Hologenome Initiative, and I can tell you we didn't observe this pattern. So,
okay, thank you.
I have no idea.
Thanks. Other question? Okay, so we are going to take a 5 minutes break.
Yep.
Okay.
Sure.
Great.
We'll get some water.
See you soon. Lo scarichiamo adesso. Vabbè, lo scarichiamo da qua. Tu l'hai scaricato? No, ho capito, l'hai scaricato, mi stai dicendo, mille cose. Ma sei sicuro? Lo devi riprendere lo scarico?
Esatto. Perché quelle non sono delle uguali, nel senso, sono uguali, però
Sì, poi alcune conservano dentro le condizioni magari, sto pensando, no, altre misure contaminanti, altre ma insomma c'è sicuramente tante cose, proprio una pietra, cioè quelli sono così rari che non posso completamente sostituirlo con un altro animale. Ah, sani fanno tanti nemici. il campo di Genesis comunque di gestione degli studi. Lei sicuramente ha fatto una fissa che di materiale o in campa, sostanza e quant'altro, selezionando una gestione sì, anche male,
Anton?
Yes.
You want to start?
Yep.
Okay, I'm gonna call the student. Okay. Please have a seat. We start. Thank you.
Okay, shall I proceed?
Yes, thanks, Anton, please.
Super. Okay, so now we will continue with the bioinformatic part of the metagenome analysis, and then we will cover a little bit of host genomics and microbial and host transcriptomics. But for the bioinformatic approach, I know that you have covered a little bit shotgun metagenomics in the morning. Have you deepened on the bioinformatic approach to analyze the data? Perhaps a little bit. So regarding shotgun metagenomics, would you be able to define very briefly mention what possibilities we have to analyze this? Did Mark mention anything about metagenomic assemblies, binning, max, etc.? No. Okay. So it was mainly based on a read-based analysis. Cool. So then it won't be overlapping, and that's very good. So When we get our sequencing data, and let's imagine now that we have pure microbial data. Let's imagine that we have this ideal situation in which we have no host DNA and no non-bacterial DNA. So the raw reads that we get from the sequencing machine needs to be filtered based on the quality. If we had host DNA, the approach we use to remove that host DNA or to split that host DNA from the rest of a metagenomic fraction is to map the reads against the host genome, a host reference genome. In this example, I have it added this because I will try to keep it simple. And I will keep it simple because the metagenomic analysis can be done mainly in following three strategies. I think in the morning, Marco mentioned a few approaches for doing read-based analysis. I think he mentioned Metaflan, perhaps. And using Metaflan, this is a read-based approach in which what we do basically, explained in a very simplified way, is to map the reads that we get to different bacterial reference genomes, and we assign taxonomy to each of these reads. This could be done by actually mapping, or it could be based on k-mers, different approaches to set the relationship between the reference sequence and the reads. But this is the simplest approach for generating shotgun, also analyzing shotgun metagenomic data. It must be said as well that it is the weakest approach. It is the weakest approach mainly due to two factors. One is that there are many ambiguities in the taxonomic assignment to short reads. When we do short read sequencing, the sequencing reads that we get are usually between 100 and 150 nucleotides long. That means that many of the reads cannot really be assigned to any taxa unambiguously, because 150 nucleotides is quite short sequence length. And these sequences could be really assigned to multiple microorganisms. And this is what it prevents us from getting very robust taxonomic annotations. The other limitation is that the read-based analyses are completely dependent on reference databases. Unless we have reference databases, we won't be able to annotate the short reads. And I didn't include here an example of a study that we have done, but when I saw Marcos' slides and saw Metaflan there, for example. So depending on, Metaflan uses a reference database, a specific reference database that is quite biased towards human-associated microorganisms. Depending on which database you're using, the results that you may get could be very, very different. And this is a limitation of the read-based analysis. So it is extremely important to know which reference database you are using and how representative it is for the microbial community you are expecting in your study. Because if we use Metaflan to characterize mussel-associated microorganisms or salmon-associated microorganisms, et cetera, it can happen that the retrieved community can be considerably distorted. That's something we have observed, for instance, with guinea pigs, in which proteobacteria are overestimated considerably. And this is nothing wrong with the actual algorithm, but it is an issue with the reference database. If the reference database has been mainly created based on human-associated and mice-associated microorganisms, then you need to be very cautious on how you extrapolate that to other taxa. Another very relevant thing here is that whenever we go for wild organisms, it is very common that 95% of the genomes that we recover are novel species. It sounds crazy, but we have cases of lizards, for instance, that we have characterized within the Earth Hologenome Initiative, and 99% of the hosts of the microbes that we detect are novel species. So we are still at that point in terms of characterizing host-associated microorganisms. There's a lot of unknown diversity, and all this diversity is not included in these reference databases. So if we don't have a complete reference database, all these short reads analysis will be very, very challenged, and it won't really give the information that we want. So this is something to keep in mind. But as I said, read-based analysis is not the best way to go ahead. Sometimes it's the only way that we can go ahead, because I will mention that, that we might have some limitations in terms of sequencing depth, et cetera, et cetera. But the best way to analyze shotgun metagenomics is to run metagenomic assemblies. So do you know what a metagenomic assembly is? Can any of you explain what a metagenomic assembly is? Or a genomic assembly? Have any of you assembled the genome ever? So a genomic assembly or a metagenomic assembly is basically a process, a procedure in which overlapping reads are converted into the so-called contigs, which are longer reads that are derived from overlapping short reads. The process of doing this is slightly different between genomics and metagenomics. And the reason is that whenever we do genomic assemblies, we expect that the coverage of the entire genome to be even. That means, let's imagine if we have 1 million cells that we have lysed and we got DNA from, the genome should be 1 million times represented, but the entire genome should be 1 million times represented. So we rely on this evenness. When we do metagenomic assembly, we don't have that assumption because different bacteria appear in different abundances. So that's the main difference that differs a metagenomic assembly from a genomic assembly. But the underlying idea is the same. We will get short reads, and whenever they are overlapping, we will try to create longer reads. There are many benefits of getting these longer reads. One of them is that as the sequence is longer, the statistical power that we get for assigning taxonomy or assigning functions is much higher. We are not anymore relying on 150 nucleotide long sequences, but we might be talking about thousands or hundreds of thousands of nucleotide long sequences. The success of assembling a metagenome depends on multiple factors. But the two most important factors are the sequencing depth that we have and the diversity of the metagenome. The more diverse a metagenome is, the deeper sequencing it will require. But there is also inherent limits that the algorithms have to assemble metagenomes. If the metagenome is extremely complex, as it might be the case of some soil samples or even some fecal samples of some of the animals, when we have thousands of different bacteria, the metagenomic assemblers really struggle to yield good assemblies. So in those cases, sometimes it is better to dilute the samples and try to reduce the community diversity in order to get some assemblies, because sometimes we will be facing real challenges. The genomic assembly with very diverse samples is really difficult. But of course, if we don't have enough sequencing, we won't have enough power either to generate these contigs and get longer sequences. Do you know what might be another benefit of getting these contigs rather than short reads from a functional perspective? If we want to get functional information, what do you think it is the benefit of having contigs compared to short reads? We can ask an intermediate question. In order to do functional inference from a metagenome, what is the unit of information you will need? The unit of information you need is the gene. In order to assign a function to a sequence, you need to define genes first, because then you can assign functions to the genes. When doing metagenomic assembly, the capacity for predicting genes increases considerably, because usually there is no gene that has just 150 nucleotides. Genes are considerably longer usually. And unless we do this metagenomic assembly and we reconstruct this context, we won't be able to predict genes and to assign function to the metagenome. It is possible to do this using a read-based approach. So this initial approach, in this case, by using genes, reference genes, instead of reference genomes. So instead of assigning taxonomy, we'll be assigning function. That is possible, but this would be facing the same issues that the taxonomic assignment is facing. So this kind of ambiguities and also a huge reliance on the reference database. But if we do assemblies and we reconstruct this context, this long context, we are able to predict genes within those sequences. And this prediction is based on the structures of the DNA. So it's not based on a reference. It is based on the structure of the gene, of the nucleotide sequences. Because we can, using starting codons, using stop codons, using different promoter structures, etc., we can quite reliably identify what is a gene within that context. And using that information, then we can do a lot of things. One approach is to use the gene-based approach, where we get the assembly, we predict the genes, and then we annotate those genes, and we map all the reads back to these genes. And that way we can have a representation of what is the relative abundances of different genes with different functions in our metagenome. That would be one approach. However, there is a limitation of this gene-based approach, which is the genes are not independent. So if we identify 1 million genes in a metagenome, these 1 million genes are not completely independent of each other. These genes are encapsulated in microbial genomes. And it can happen that 2 genes that are in 2 different microbial genomes might be doing 2 completely different things. Even though they have the same sequence, the surrounding genes will also dictate when they will be expressed and what kind of functions they will have. But that's why the assembly plus gene-based approach itself is not either the best approach to do shotgun metagenomics. Let's say that the gold standard is to try reconstruct the actual bacterial genomes. And this can be done through the binning process. What the binning does is basically it gets all these contigs that we have reconstructed, and then using statistical methods, it creates bins that represent genomes of bacteria that we can characterize based on relative abundances of these contigs, as well as the nucleotide properties of the sequences. So it is possible to rather accurately to reconstruct bacterial genomes from metagenomic data. So what we would be getting in the end would be a draft genome of a bacteria. If we have 1,000 bacteria in a metagenome, we will probably do not manage to recover 1,000 draft bacterial genomes, but we might be able to recover 700, for instance. That's a lot of information that we can use for functional inferences of the microbiome. These draft genomes can have different quality. And this quality is usually assessed by assessing what is the completeness of the genome and the contamination or the redundancy of the genome. And this is assessed by using single copy core genes The single copy core genes are genes that appear just one time in each bacterial genome and that appear in most of the bacterial genomes that are known. So using these genes, using the presence of these genes, we can assess how complete the bacterial genome we have reconstructed from the metagenomic mixture is. This method has some limitations, but this is the best way that, let's say, the standard way that is used nowadays to assess what is the quality of the genome. If we manage to reconstruct these bacterial genomes, then we can also annotate what are the genes that each of the genomes contain. And using that information, we can create this kind of functional annotations at the MAG level. So this, the MAG is the draft bacterial genome. The MAG comes from metagenome assembled genome, MAG. And this is what we call, to these draft bacterial genomes, we reconstruct from metagenomic assemblies. If we do this kind of functional annotations, then each of these rows that we can see here is one bacterial genome, and each of these columns in the y-axis, so in the x-axis, are different functions. So it is possible to have a really huge matrix of functions and bacterial genomes that enable us to do a lot of very cool stuff. Because we can really and certainly know what are the functional capabilities of the different bacteria, not based on reference genomes, but based on our own data that we have generated. So this is why it is considered the gold standard. If we have this information, then we can create these genome scale metabolic networks, which show us what is the capacity of our microbial community to produce functions, to generate some specific metabolite, to create some other kind of chemical compound, etc., etc. In this case, this is an example of the microbial communities that are or are not able to generate some metabolites. So using this information, we can know whether the microbial community that we have will be able to produce some specific short-chain fatty acid or some specific peptide, etc., etc. And this is really powerful because we can really work on the functions and we can foresee what will be the effect this microbial community will have in the host. And that's why this is considered the gold standard. All these procedures are improving all the time. So far, I've been mentioning only short read sequencing. And you might know that nowadays there are also long read sequencers, PacBio sequencing, nanopore sequencing, and Illumina will also launch their own long read sequencer in the second half of this year. And that will also change quite a lot of things, because when we have longer reads, then we don't rely that much on the metagenomic assemblies. And the quality that we recover, the quality of the max that we recover can be much larger. So in the future, all these procedures will be polished quite heavily. So what we need to keep in mind whenever we generate data, we analyze data using read-based, gene-based, or genome-based approaches, is of course see what's our research question. Whether we are, for instance, more focused on getting taxonomic information or functional information. We will also need to assess what's the funding capabilities, because doing genome resolved metagenomics, so the MAG-based analysis, the last one that I have shown you, requires quite a lot of sequencing, more than the others. So it is more expensive. But also we also need to consider the biological features of the system that we are analyzing. Because for instance, if we will be relying on gut contents that have a lot of host DNA, then we might not be able to reach the amount of metagenomic sequencing depth that our approach might require. So based on that, we will need to maneuver and use different approaches. Do you have any questions about these bioinformatic approaches? Shall we continue then? So now we will jump from the host, so from the bacteria or the metagenome, the microbial community, to the host. And we will do that by showing this huge contrast initially. And this is the difference of the genome size between bacteria and their hosts. These are just approximate numbers, but just to show you what is the huge contrast. An Enterococcus genome is about 3 megas long, while the salmon genome is over 3,000 megas long. But this is also related to the asymmetry that I mentioned in the beginning about the hosts and the microbes. It's not just the size, it's not just the timing of, it's not just the reproduction. The genome size itself is also extremely different. So the way that we will analyze this will also be very different. What we usually try to do with the host genomic data is to create SNP profiles. In order to do genome-wide analysis, what we do is what we aim for is to create this kind of SNP profiles to show what are the genetic differences between the individuals that we'll be analyzing. This is what we do when we work with animals belonging to the same species. If we were talking about species, to multiple species, this would not be the approach that we would be using. Probably what we would be using, it would be a phylogenetic approach in which we build the phylogenetic trees and then compare the phylogenies. Or it could be also a functional approach where we create some synteny alignments of different genomes and try to see what are the functional differences on key genes. But in the next minutes, I will focus on these inter-individual differences mainly. The way we generate this data is very similar to what I mentioned. What we do is to map the reads, the sequencing reads, to a reference genome. And that requires a reference genome to be available. If you don't have a reference genome, the approach is quite different. But nowadays, we have more and more reference genomes of more and more host species. And I want to explain what's the way to generate these reference genomes, but there are many international projects and initiatives that aim at generating high-quality reference genomes for a range of host species. Once we have a good quality reference genome, we can just map the reads, the sequencing reads we have to that genome, and that will enable us to recover a SNP profile that we can use for comparing individuals. The problem is that often we don't have enough sequencing depth to properly characterize the SNPs across the entire genome. And in those cases, genotype imputation is quite relevant. I don't know if any of you have ever done a genome-wide analysis in invertebrates or in invertebrates, but in order to cover the entire breadth of the genome, will require a lot of sequencing. Because when we do the sequencing, even though that we should be expecting that the sequencing would be covering the different genomic regions evenly, there are always some imbalances. So typically we will need between 10, 20 times of sequencing of each genome in order to get a very good representation. And that's quite a lot of information that we need. So because if we are talking about genome like the salmon, that it's about 3 gigabases, we would need to generate 60 gigabases of data per sample in order to reach an 20x sequencing depth. And that's a lot of money, a lot of money. This genotype imputation enables us to somehow improve that limitation without going for so much sequencing. Because genotype imputation relates on the fact that gene locations or loci are not independent to each other. When a new generation is generated, so the recombination of the chromosomes does not happen randomly. So not all the positions are randomly mixed. There is some linkage across the different genomic regions. Usually genes that are next to each other will be recombined together. And using that information, we might be able to reconstruct missing information in the genotypes. And this is what's called the genotype imputation, which is to get a reference panel of very well characterized genotypes, and then use that information to reconstruct the missing genotypic information. This approach is especially interesting when we have a population that has a limited genetic diversity. This could be, for instance, fish that you might have in a piscifactory. You might be growing sea bream in a piscifactory, and you have animals that are genetically very similar but not identical. So if you characterize a few of these individuals heavily, then you can input that information to the rest of the individuals that are shallowly sequenced, and you will be able to reconstruct their genotypes fairly well, but just using a fraction of the money. And that's really powerful approach to get good quality data in an optimized way. There are different ways to do this genotype imputation, but this is the general idea. You don't need to sequence everything deeply in order to reconstruct genotypes. And this is mainly true when you have populations that don't have a huge genetic diversity. The higher the genetic diversity, the larger will need to be the reference genotype pool that you use in order to do that imputation. That's why it is feasible for farm animals. It could be also feasible for a restricted population. But when you do world scale genomic analysis, then it is quite challenging, because if there is a huge diversity, then you need really to represent all this diversity in your reference panel, and that's not easy. So, do you have any questions about the genomic approaches? Is any of you doing whole genome sequencing in your project? No. Okay. So in the future, if you will be planning to do that, this is something to consider. It is not necessary to do deep sequencing in all the individuals. So it's worth thinking a little bit about the strategies to optimize this, because you will save quite a lot of money. Okay, so now we will jump to the next step, which is another layer of complexity in the data that we generate. So when we have a genome sequence of a host, that's the top line, or multiple genome sequences of the metagenome, the microbial communities, we have another level of complexity that we can add here. And that's the gene expression. Both eukaryotic hosts and prokaryotic bacteria have genes that are expressed, and this gene expression can be used to polish the functional information we get and to get a much more refined information on what are the functional capabilities of hosts and microbes. And that enables us to much better disentangle these interactions that happen between hosts and microbes. But gene expression analysis are challenging. In general, genomics, metagenomics, hologenomics is challenging. But gene expression analysis are especially challenging if you use shotgun approaches. Targeted approaches using qPCR is quite straightforward, but when you do RNA-seq or whole genome or whole metagenome RNA analysis, it is not very straightforward. And it requires quite a lot of thinking. Is any of you doing any RNA-seq analysis in your PhD projects? Did I hear a yes somewhere?
RNA-seq.
No.
Sequence of RNA, no.
Okay, so I will give you some highlights, some hints on how to optimize this and for your future projects. So when we do gene expression analysis, usually of whole genome gene expression, one thing that we often need to do is to assess what's the quality of the RNA extraction. Because as you might know, messenger RNA and ribosomal RNA is degraded quite fast in the cells. So messenger RNA and ribosomal RNA is expressed very abundantly, but then there are RNAs that degrade the RNA very fast. Because otherwise, if the RNA accumulates in the cytoplasm, then the bacteria, the cell can just be crazy because it doesn't know what to do or has to do everything at the same time. So RNA is very sensitive to the sampling time and to the preservation. And if we don't sample the animals very fast and we don't preserve the RNA properly, RNA degrades very fast. And this level of degradation can be assessed using these RIM values, which is the RNA integrity values. Whenever we have good quality RNA extract, we should be observing something like this. This is a eukaryotic RNA extract. And I can know that because it has an 18S and a 28S ribosomal subunits there. And that's specific to eukaryotes. Prokaryotes don't have the 18S and 28S, have 16S, 23S, 5S. So there are different ribosomal subunits in prokaryotes and eukaryotes, but the idea is the same. Ribosomal RNA is expressed much more abundantly than the messenger RNA or the microRNA. So if the quality of the extract is that good, as in the first example, we should see these two peaks. In the case of the of the host transcripts, is the 18S and the 28S. In the case of prokaryotes, we should be expecting the 12S, 16S, and the 23S. So there are differences. If the quality is degraded, then these peaks disappear, and everything is much more degraded. And that will give you an idea of what will be the possibility or the capacity to recover valid information for the RNA. Because you will be getting anyway RNA sequences, but whether this is representative of the cells that you sampled will be another thing. So it is important to assess these RIM values to know whether you were able to capture and preserve the RNA of interest. The funny thing, though, is that when we do RNA sequencing, we are not interested in the rRNA. We are not interested in these peaks of 18S and 28S ribosomal RNA, because these sequences are identical. So it is expressed millions of times, but it is redundant information. We don't want that. What we usually aim for is the messenger RNA that is far less expressed than the ribosomal RNA. So it's there in the bottom. I have just this pink square. What we are interested in is that bottom. We don't want these peaks. These peaks are just to assess that the quality is good enough. We don't want that. But the way that we use for doing this is that whenever we do, say, RNA-seq analysis, the RNA itself cannot be sequenced. So the RNA cannot be built into sequencing libraries. We need to have DNA for that. So what we need to do first is to reverse transcribe the RNA into cDNA, into complementary DNA, in order to then prepare the libraries and do the sequencing. So there are two different ways to get rid of these rRNA peaks. And these are different for eukaryotic organisms and prokaryotic organisms. And the reason is that the basic biology of gene expression in the eukaryotes and prokaryotes is different. I must say that these RNA sequences, if you don't get rid of them, they can take over 95% of the RNA sequences. So you might end up with just 5% of your library being indicative of anything that you want to know about the functions of the genes of the bacteria and the hosts. So it is extremely important to get rid of these topics. The reason of having these two different approaches is that eukaryotic organisms, whenever they transcribe their genes, the messenger RNA has a poly-A tail, has an end of a few adenines in the end, that it is very handy for the cDNA conversion. The prokaryotes do not have that poly-A tail. That means that if we have this poly-A tail, when we do the cDNA conversion, we can use some primers that contain this poly-A tail, and you will only convert into the cDNA the sequences that have that poly-A tail. So anything that is eukaryotic messenger RNA will be converted to cDNA, but the rest will not. And that's a very powerful way to get rid of ribosomal RNA, because the ribosomal RNA doesn't have the poly-A tail, but the messenger RNA does. But this only happens in the eukaryotic organisms. In our case, would be only the hosts. If we want to generate data from the prokaryotes, we cannot rely on that. So whenever we have this prokaryotic, we are interested in the prokaryotic RNA, what we need to do is to create a subset of the library. And there are two ways to do this. One way is to rely on hybridization, in which we have some guides that are able to bind to the ribosomal sequences, and we'll remove those from the pool. The other option, the newest option that we are using, is to rely on CRISPR-Cas9. And using CRISPR-Cas9, what you can do is whatever you can create the same as guides, but you can tell the Cas9 protein, destroy all these sequences that have this sequence. So you can tell the Cas9 protein, anytime that a specific sequence, which in our case will be a ribosomal RNA sequence, is there, destroy it. And this way you can get rid of the RNA as well. And then you would get much more pure sequences, RNA sequences. Usually you don't manage to get rid of the entirety of the ribosomal RNA, but you are able to enrich the library considerably. And rather than having 95% of ribosomal RNA, you might end up having 5, 10, 20% of ribosomal RNA, which is a big difference in terms of money. When we go for these multi-omic approaches, and when we don't just do genomics and metagenomics, but also transcriptomics or metatranscriptomics, we have also the possibility of using mass spectrometry-based analysis, which could be the proteomics and the metabolomics. I won't go beyond just mentioning this, because I don't have myself the experience of generating this data. I have started to analyze proteomic data and metabolomic data, but not producing it myself. So this is just so you know, we enter into a completely different domain. This is not based on nucleotide sequences anymore. We are not talking about generating DNA and RNA sequences, but we are talking about using mass spectrometry to identify structures of different peptides, of different metabolites. And these structures and the identification of these structures is completely also dependent on the reference material that we have. So you will need to use some standards, and then you will need to compare the peaks you get in the mass spec to the reference material that you have. One of the main limitations of proteomics is that usually you manage to retrieve just a fraction of the expected proteins. So, and it is very dependent on how you prepare the structs, because you can do mass spectrometry with liquid chromatography or with gas chromatography. Depending on how you prepare the extracts, then you might be able to capture metabolites or peptides that have certain properties, certain polarities, solubilities, etc., while some others will be lost. So it is not easy to span the entire diversity of proteins and metabolites that you might have in the sample. So usually it is necessary to target a little bit. So target to peptides or to metabolites with certain physicochemical properties, rather than spanning all of them. So this is something to keep in mind. And then we have the very challenging part of the data integration. And I won't deepen into this because it would be too complex. And it is something that it is in a extremely dynamic phase, because we have been able to generate a lot of multi-omic data. But multi-omic data is complex by itself, but it's also very let's say, non-statistics friendly, because whenever we generate multi-omic data, usually we have very small sample sizes and we have a lot of parameters. So, and this is not good for statistics. And this is a challenge that we'll be always facing whenever we do multi-omics until we reach a stage in which we are able to generate this data for thousands and thousands of individuals per study. But we are not there yet. And I guess that we won't be there in at least in the next 20 years. So it is quite necessary to develop new tools that enable integrating all these different types of omic layers in a statistically sound way. And there are a few projects, and I am involved in a few projects that aim to do that. But the main idea is that we need to somehow simplify the complexity of the data sets that we generate, try to reduce the number of parameters that we'll be using. And this can be done using the different multiple ways. But once you manage to do that, and once you get enough statistical power, then the amount of information you can get it's amazing. And the kind of inferences you can do, it's really great, because you really have a very systematic picture of what's going on. And it enables you to really understand what are the interactions between the hosts and their microorganisms. So it is 6 already. Do you have any questions? I am done with this third session. Would you like to ask anything?
Any question? No.
No? Shall we continue then?
Yes.
Good. Last session. And this session will be mainly about things to consider when applying hologenomics into any study that you might be considering for the future. So this would be the question that I think if you are considering ever using hologenomics for addressing any scientific question, it's when and how should I implement hologenomics? When does it really make sense to go for hologenomics? And this is a very pertinent question to do, because hologenomics is complex and hologenomics is expensive. So it is really necessary to think a little bit and to ask ourselves some questions that are very basic questions. And I prepared these 5 questions that I consider that is relevant, that each of us should ask whether does it really make sense to do hologenomics. The first question would be to assess what is the complexity of the study system, because we might end up realizing that the complexity is too high, or we might end up realizing that the complexity is too low, and perhaps it does not really make sense to do hologenomics because there is no complexity in the host genome. If all the genomes are the same, then there is no point in doing hologenomics, for instance. It is also important to consider which variables can be controlled by the researcher, because unless we are able to control something, it's going to be extremely difficult to get any inference. If we are unable to control anything at all, we might be able to find some correlations, but we won't be able to really get any causation on any causal relationship on the analysis that we do. It will be also relevant, as I mentioned at the very beginning, that to assess what is the resolution at which the hologenomic variation will be studied. When we do the host analysis, are we going to do whole genome sequencing, or are we going to just rely on mitochondrial genomes, or are we going to just use one gene and we will rely on one gene? And the same with the microbiota. Are we going to use amplicon sequencing, or to use, are we going to use entire microbial genomes? So it's going to be necessary to think about all this. We will also need to think about the spatiotemporal features of the studied hologenomic variation. So how often do I need to sample? What is the resolution that I need to sample? Is it enough to get fecal samples? Should I get the entire intestine and characterize the entire intestine? Should I get just one part of the intestine? And in that part, should I get everything or just the mucosal barrier, or just the digesta. So thinking about this is essential. And the last one would be also to think about the statistical part of it and start thinking about what are the explanatory and the response variables. Because I will show you that hologenomics can be applied to different statistical designs in the end to address different questions. So one figure that we added to this paper was this one that represents the complexity of the three main angles that we need to consider when doing hologenomics. One is the genomic complexity, how different and how complex the genomes that we are analyzing are. The metagenomic complexity, considering how complex the microbial genomes that we will be analyzing are. And then the environmental complexity. We need to know whenever we are comparing individuals, whether the environmental factors that are linked to these individuals are different or not. So depending on what is the level of complexity, we might be able to do some things or other things. So if we go from bottom to top, this is an increased gradient of complexity. So for instance, if we work with inbred and germ-free models, we will have a genomic complexity that is extremely low, because if they are inbred, they will be genetically almost identical. If we are talking about germ-free models, we will be able to inject the microbial community that we want, so we have a very low complexity of the metagenome. And if we are in the lab, we might be able to control the environment very much. But if we go to the other extreme, we are comparing wild species, then everything will be extremely complex. So considering this in the very beginning is extremely relevant, because this will enable us to answer different questions. If your aim is to learn something about how toll-like receptors affect the detection of some specific MAMP and blah, 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 then don't go for wild species that you get in the environments, in the wild environment. That will not be the resolution that you will require to address that question. But if what you want to do is to assess some evolutionary relationships between host evolution across different taxa and microbes, then don't rely on lab animals, because this won't be representative of what is going on in the wild and in nature. So different questions will require different levels of complexity. And this was represented here in this other three-dimensional box, where we have the environmental complexity, metagenomic complexity, and genomic complexity that different types of studies have. And that will enable us to address different questions. If we go to the second question that we need to ask ourselves, is which variables can be controlled by the researcher? Of course, if we go to the lab and we get animals that belong to the same family, or animals that belong to the same genetic line, we will be able to control the genetic variation. If we go to the wild and rely on wild mice, for instance, we won't be able to control the genetic variation that we will observe that much. Because we might be able to control, for instance, by contrasting one population from Italy versus another population from Spain, for example. We know that genetically will be two different entities, but we won't really be able to be more detailed, because we want to really know what are the relationships that each individual has, which the rest of individuals that we sample in the field. When we go for the microbiome, pretty much the same. If we are using synthetic communities that we are injecting to germ-free animals, that will be very much controlled. If we do antibiotic treatments and then add gut microbiome transplants, then we will have some level of control. If we are just characterizing microbiomes from wild animals, we won't have control at all. So that will enable us to answer different questions. And finally, we have also the environmental factor. If we are working in the lab, we'll be able to control most of the environmental factors. If we are in the field, we won't be able to control most of the factors. But at the same time, the representativeness of the environmental conditions we will have will also need to be assessed.
what is best, to have a very controlled environment, but that does not really represent the natural reality? Or is it better to have something that is less controlled, but it is more representative of what's happening in the wild? So depending on the question, we will need to go for one or the other. This is linked to what I mentioned in the beginning, where we can select what is the resolution at which each of the parts of the system will be considered. So in the case of the host, we can do whole genome sequences, as I mentioned, but we could also base our analysis in a single marker. And depending on which question that we are trying to answer, it might be good enough. With the microbiota, the same. We can do 16S sequencing, or we can do a MAC-based analysis in which we reconstruct the genomes of the bacteria. And environment-wise, we can also have the same, because there might be some environmental factors that we might consider, and might be some others that we do not consider. And depending on how we define this, will be relevant or not. Then if we talk about the spatiotemporal features, we will need to consider how often, so when, how often, and for how long we will sample the animals in temporal scale. And we'll also need to consider the spatial features that I mentioned earlier. Are we gonna collect the fecal material? If we go for the fecal material, do we consider this is representative of colonic microbial communities? Are we interested in the colonic one, or we want a representation of the entire intestine? So we will need to consider that as well. And finally, we will need to consider what are the relationship between the explanatory variables and the response variables. So in many cases, and mainly when we are in the production context, we might want to model what is the phenotypic response, or what is the performance of the animal when we equate the genomic properties with the metagenomic properties of the environment. And sometimes the question might be different. We might be interested in how genomic features, expression of genes, for instance, or methylation of the the genome is affected by the combination between the metagenome and the environment. Or it could also be the opposite, how the metagenome is affected by the genome and the environment. So depending on what we are planning to do, we will need to ask ourselves all these questions. I know it is complex and requires a lot of thinking, but it is really something that it is worth doing before starting analyzing or creating a study design. So we are approaching the end. My question for you would be whether you can think of any research questions you think could be tackled with hologenomics. I know we are all tired. It's been a long day. But I will give you 1 minute. Let's see if one of you can have any suggestion, and then we can be happy and we can happily end before time. I'll take 1 minute, think about this, and try to also think on your own research context, whether hologenomics would make any sense. And hopefully we can discuss a little bit about this. Take 1 minute. I will mute myself for 1 minute. So,
Please.
does any of you have any active neuron?
Do you hear me?
I was asking whether any of you have any. Yes.
We
Super.
investigate, for example, how human disease are correlated with microbiota in different populations, for example. Yes, if there is a relationship between also the phenotype of a population and the microbiota.
Mm-hmm. That would be one possibility, for instance. Yeah, so that would be linked, so perhaps to this, right, where we have a phenotypic response that could be a pathogenic response that the host might have for some detriment, and how this is affected by the genotype and the metagenotype. It could be also this one, right? How the metagenome that's different populations are showing is linked to the environment in which they are living and the genetic properties of the hosts. That would be one option. So in that case, what level of resolution do you think you would need for characterizing properly the host genome? Do you think it would be enough to select one gene of one human gene and sequence that, or would you require whole genome sequencing of human genome?
I think it depends on what we are looking for. For example, for a disease, if we know the mechanism of the disease, but probably not only one gene, but a combination of genes. I don't know if the entire genome, but.
Mm-hmm. No, yeah, exactly. If you have already a suspected association and you can narrow down the analysis to a gene or to a set of genes, the analysis would be much more, would be faster and cheaper, for sure. Of course, if there are other aspects, other factors that are also contributing to this, then you will be missing those. But it will be all about finding the trade-off and the most optimal way of generating the data and getting the information. Okay, so that would be one example. Do you have any other example from your own research that you feel that would be relevant to implement this approach?
This morning, I take inspiration from Marco class, for example, the acclimatization potential of a population, marine organisms to climate change, for example.
Cool. Okay. This is one of the examples that I added actually here. The third one would be the joint means of adaptation to environmental change. So we could analyze what are the genomic signatures and the metagenomic signatures of adaptation to climate change. And this is something that we are actually doing in the Earth Hologenomics Initiative. We're using terrestrial vertebrates, but we are analyzing how the joint action is potentially enabling animals to adapt to environmental change. Could be a climate change, but could be also adaptation to urban environments, or could be other type of or to polluted environments, etc., etc. There's another interesting area, which is the hologenomic means of domestication, or to say conversion of wild populations into production animals. And there are quite a lot of examples in marine organisms, in fish mainly. A lot of research groups that are trying to see what is the most optimal way to maximize production based on wild organisms. And so considering both host genomic and microbial metagenomic information, it's quite relevant to find the most optimal output there. Then is, of course, the ones that are already domesticated. Could be salmon, could be trout. We have multiple projects in which we try to improve the production based on these host microbiota interactions. And we have a rather cool example in our ongoing European project in which we are trying to see what is to understand what is the response of the microbiome and the host jointly to different sources of proteins for growing salmons. As you might know, salmons are originally carnivorous and they require proteins. And so far, these proteins have been derived in part from fish. And it is quite unsustainable because nowadays, for instance, the Norwegian salmon industry is using fish meats of fish captured in the Indian Ocean in order to feed salmons in Norway. So in the sequence, in the process of finding more sustainable ways, we've been exploring the possibility of using blue mussel proteins instead that can be produced next to the salmon tanks and gets the amount of animal derived protein that the salmons require in a much more sustainable way. Of course, proteins derived from fish are not 100% similar to the proteins derived from the mussels, and this entails some changes in the host reaction and the microbiota reaction to these proteins. So we've been analyzing those, for instance. And there are many other examples of this way in the production context that I guess will be also relevant for you in the PhD, in this PhD. I don't know if you have any other questions, any other comments you would like to mention before we finish.
I have a question. Maybe it's a challenging one because it really depends on the question of the study, your goals. But in general, what is the sample size in your studies? Sample size of wild organism, for example.
I can tell you what
the biodiversity, the biota of a population, a wild population. How many sample size are you using in general? Just an order of magnitude.
Yeah, as a rule of thumb, in the Earth Hologenome Initiative, we are using 10 individuals per population. 10 individuals per population. And then depending on what we are trying to address, each species might be represented by between 10 and 20 populations. It depends on what we are aiming to do. But we are talking about at least 100 individuals per species in order to capture a representative sample in order to reconstruct this metagenomic space in a trustworthy way. Of course, this is very dependent on what is the ecological niche of each species. So for instance, if we are trying to understand what is the microbiome variation of the house sparrow, which is present in the entire planet, basically, compared to another species that is present in a very localized area, the sampling size that we will need will be completely different. And the same happens with any fish species that we might want to sample. But I would say usually 10 individuals is rather good based on our experience to characterize properly a community, always trying to standardize the rest of the features as much as possible. So developmental stage, sexual stage, status, sex, etc., etc. The more we standardize those and that we limit the variation, we will need less samples. But of course, with 10 individuals, you are unable to capture the entirety of the diversity, but it's usually good enough. Anything that is less than 10, it will be quite limited, I would say.
Thanks. Any other question?
I'll stop sharing this.
Okay.
Well, if there are no other questions, it's been a pleasure from my side. I know this is quite challenging. I would rather prefer to be there, and it would be much more optimal for interacting, etc. I have also almost lost my voice. So.
Anton, thank you very much from all of us. Marco is here. Maybe you want to say something.
Likewise. Thank you so much for the invitation.
Thank you.
And hopefully the next time will be in person.
Yes.
And, yeah. So.
Yes, next time will be in presence. Okay. Thank you, Anton. Thank you.
Thank you very much.
Bye.
Have a nice evening. Goodbye.
Bye. Can you close the record? Ciao, Anton. Thank you. So, okay, so for today we are done. We meet tomorrow morning 9:30. We have sorry, I'm John. Okay, I'm finished. Tomorrow morning we have Gianfranco Uggen, University of Bologna, talking about crystal structure, and then in the afternoon we have Daniel Strali online from the Hologenomics Institute in Croatia. Professor Giovanni Valdre will master the section here, and I will be connecting with you from the main campus because I need to stay there tomorrow because I'm teaching to another PhD program more afternoon. So tomorrow will be with you, Professor Valdre. So I see you tomorrow morning, 9:30. Have a good evening. Bye. Thank you. Bye.
Coppa computer.
Yeah.